Health Education Summit. We are very excited to be bringing you this content. I have been uh, supported by an extraordinary array of uh, researchers and assistants here on this project. This project has really been a part of my last 10 years. It happens to that our scientific efforts of that last decade all became super pertinent in this last few minutes of human history. And so what has felt like a sudden experience for the world felt like a very clear next step of education and scientific research, as well as now public health research in the areas of viromics, the microbiome, and human health at large. And so it is my pleasure to try to compress that decade of information into the next couple hours here. And it's going to be an intense time because there's a lot of emotional content within this. The emotional content during this period of time, when we have so much going on in the world, is underlined by the extraordinary human impact that the last year and what we've called the pandemic, and both the viral experience, the viromic, story that we've told, as well as this bigger story of human health in the context of our socioeconomic collapse, the fragility of our nation. So many topics like this have put an incredible amount of stress on the population. So as go through this content, there's going to be moments of real frustration and real anger that would be natural to all of us when we realize how little we actually know and how far from the truth science and its narrative currently has us. And so I want to prepare us for those moments of frustration, those moments of anger, those moments of hopelessness, those moments of perhaps fear. And I want to shake that all off, because the worst thing that could come out of this is the experience of leaving you with a sense of instability or a sense of more questions than answers. I think that on some level, the questions will be more than I can ever answer. I think that on some level, there will be important questions that will never be asked in the public narrative of what's happened this last year. But I want to free us up with the possibility that our real purpose here together is not so much to be whistleblowers, point out the mistakes that have been made by doctors, scientists, public health officials, politicians, or the rest. It's really about envisioning a different future. And so my concern of the last 6, 12 months has waxed and waned on a near daily basis. Some days I am in apoplectic frustration with the situation and anger at the powers that be. And other times I'm just blown away by the resiliency of the human spirit, and also recognize that this last year has been a massive reset to humanity as a whole. And we needed this reset on a deep level. We needed it psychologically, spiritually, and in fact, I think physically and in this medical realm, we needed this reset, this new reality to set in. And so I'm eager to share with you many aspects of kind of what I see happening in this near future in regards to where we could go as a population, where we could go as a group of scientists and physicians, where we could go as a species. And so this is, I hope, really what you continue to touch back on throughout this is we will do very well for ourselves if we learn rather than fear the past, if we learn from and anticipate more learning from the future than what we would fear from it. And so I hope that you are intrigued by anything new that you hear today. And I hope that it starts to open up your eyes of the ways in which you could participate, your family could participate, your community could participate in a different reality, a different future for humanity, and particularly for our children. We want a different reality than we've lived in this last year. And so I'm going to dive into some of this, data in detail, but I want you to know that I can't give you the service of the full dive on any of it because there's so much to cover. And I'm already afraid that I'm going to go a couple hours strong here, just getting through the content that I've tried to distill down to its most substantive overview. Each of these subjects, of course, could be dived into in great depth. And over the coming years, we will continue to try to do that for you in these months ahead, years ahead, with the Global Health Education Summits each month, as well as our continued ongoing education through Farmers Footprint, our nonprofit, as well as the science team at Ion Biome and the Ion Biome products that continue to kind of push the paradigm of our understanding of how human health occurs within the context of this great microbiome. As a starting point, though, I do want to recognize that it's worthwhile to realize that what has happened in this past year has been extremely unique in that we are finding ourselves at a tipping point, not just of human history in regards to a pandemic or disease patterns, but of consciousness. We are in this massive philosophical, scientific, and I would say consciousness shift for humanity that I think is very hard to find another time in history that equates to this period and to the kind of revelations that are within this period that might be marked by a couple of decades. 2,000 years ago, we could say that a similar event occurred in that we had the modern geometry discovered by the Greeks, and Pythagoras and his colleagues came up with an extraordinary discovery that the earth was not flat, and in fact it was a sphere. And that was such a revolution in human context because it made no sense with our current understanding how we weren't falling off the planet. Somebody standing on the bottom of a spherical planet would have to fall off. We didn't understand the possibility that we were a spinning globe with a dynamic gravity that would be holding life together in this spherical planet that we live on. A change that big took centuries to take a hold. And of course, it's not without some degree of concern that we find that today we have a pretty vibrant flat Earth society that are trying to make scientific, reality around the possibility that the Earth is not at all round, but it's ultimately flat still. And so when you have these cataclysmic paradigm shifting sciences, it can take, in this case, millennia to really bring about universal adoption of this new idea. And so when we have these big shifts in science, big shifts in the perspective of humans, we can expect time to be a necessary element within the adoption and the new questions that need to be asked, the new answers that need to be discovered to lead us to the more profound questions underneath those. 400 years ago, Galileo discovered the telescope, or colleagues of his, and it was his application of that telescope that really gave us the next major shift. Now, not only was the Earth round, it was no longer still. He discovered that we were a tiny rock hurtling through space and orbiting the sun, and that sun orbiting through a large galaxy, and that galaxy spinning out into the universe at large. And so that massive cataclysmic, shift in science again 400 years ago was such a humbling thing, not just scientifically, but even on the religious level, for us to spiritually come to the conclusion that we were one planet in more than 1.5 billion planets within our tiny little galaxy, and that there was a couple billion, if not hundreds of billions of other galaxies. The numbers got so vast so quickly over those hundreds of years that we have had to really struggle with who are we, why are we here, and where are we going? Are we alone? These big fundamental questions about the universe. And we're just 400 years into the understanding of that place. We've been around for 200,000 years as Homo sapiens sapiens, and we're just starting to scratch the surface of these extraordinary questions. We are no closer to an answer scientifically or spiritually as to, are we alone? Are we here to participate in a galactic community of species throughout the galaxies? Or are we here as some sort of manifest destiny of biology itself on this tiny little rock in the middle of space? There is no consensus in this space after 400 years. But there's something even more profound that's now just uncovered in these last decades, and that's that the human cell is not at the center of human health or human disease. This is so disruptive to our understanding because now we're not just challenging our planet and perhaps the role of our species on that planet. We're starting to challenge that origin story of humanity and perhaps where we are going and why we are here. To discovery that the microbiome and the virome, the vast viral information that we swim in, is in fact the mechanism by which life has occurred on the planet. And beyond all other things, certainly the mechanism by which it has been possible for mammals to develop and ultimately humanity to develop into the conscious beings that we are today. The idea of how many mistakes we've made in the last 100 years alone, studying human cells in sterile petri dishes, coming to stark conclusions about how cardiovascular disease works, or how cancer works, or how infectious disease works. All of this stuff determined into its hard belief system within these sterile petri dishes. No contact with vast microbiome information streams, no contact with incredible enzyme and protein synthesis capacity of that microbiome, bacteria and fungi alike, blowing our minds in their capacity to support human health. We have so sold ourselves short by this myopic kind of manifest destiny of the human belief that we were the most important and most advanced biology on the planet, to find out that we are fully dependent upon, and in fact, probably coming from the the most humble among us, these microbes that are invisible to the eye, living within our soil, water, and air systems. This is the revolution we're in right now. And so as you look through all of this data about what's happened over the last year, I want you to give some grace to the scientists, to even the public health personalities that have so dictated the narrative that we've been under and the rest. Let's give them a moment of grace in recognition that they have built their rigid philosophy and belief systems on thousands of years of beliefs about human biology as some sort of penultimate biologic experiment of the world, or perhaps of a God out there in space. They have a very rigid belief, because it's been reiterated so many times in their experience, that humans are their own entity. They are an isolated species that are capable of battling against invaders, and they maintain this warlike philosophy of the germ theory and everything else, because it's been told so many times, because we've studied everything in the context of these old belief systems, that the human cell was at the center of human health. Of course it would have to be. And so a measure of grace to myself as a physician, to all of us as physicians, to all of us as scientists, to all of us as public health experts. The reason we are so ass backwards on so many things this year is because the paradigm shift is so new. We are struggling to understand, what does the world look like in the context of a world in which the microbiome and the virome are at the center of human health. It's a struggle for all of us. It takes time to ask the right questions. It takes a longer time to get the answers to those questions, and a much longer time to adopt those answers into a new understanding of who we are and why we're here. And so as we begin, I would ask for the grace to be there. Another thought right before I jump in here that I want to cover is that science is not defined as a body of knowledge. It's very important. And this gets at, again, why we're so capable of something that I would call scientific dogma, which are two words that should be impossible to put together. Scientific dogma is, in fact, I think, very much what we've seen dictated to us through the narratives of the media and the CDC, the WHO, the NIH, all these big regulatory bodies that have come forward to give us some sort of cohesive narrative, have been using the concept of science as if it was a body of knowledge that was finished, right? That was, here's how many cases of COVID there are, here's how many deaths there are, whatever it is, and then that's the science. And anybody who disagrees with the science of that, or the science of, you know, masks or social distancing or vaccines or whatever it is, are not only, you know, ridiculed, but now in the British Medical Journal just yesterday, an article suggesting that perhaps anybody that's in opposition, any scientist or physician in opposition to the narrative of the importance of vaccines, should be criminalized. That behavior of this, what they call in the article of British Medical Journal, anti-vaxxing, should be criminalized. And so I want to point out to you all and to the world that science is not a finished body of knowledge. It's not like a library. Science is in fact defined as an activity, an exercise of intellect and methodology applied to advancing understanding. And I want to point out the difference between that definition, which uses the word understanding, and the word knowledge. We are advancing our understanding of the reality that we live within, But at no point does science believe that it has finished the journey and we suddenly have that deep knowledge and we're done and we know everything. And science would be irrelevant if we had knowledge, if we had true knowingness of everything in the universe. Science wouldn't be necessary. Science is the advancement of our understanding of what is real around us, and it is a process that is always changing, always advancing. And so again, when we have dogma, or when we have an old narrative that's based on 100-year-old science, of the belief of germs that are attacking humans and viruses that are attacking humans and the rest, we should give grace, but we should also acknowledge that that story cannot stay this stagnant. That story has to respond to the last couple decades of genomics, showing us that the human cell is not at the center of human health, but the microbiology and the virology around us is what defines us. And so science is a moving body of knowledge that should always be changing. Anybody who resists change in that body of understanding should be highly suspect. Anybody who resists change of our understanding of the world that we're in should be recognized as somebody who is behind the eight ball, who has lost track of the very purpose of science, and I would say the very purpose of curiosity in the human spirit. We should expect our knowledge to change. We should expect our understanding of that knowledge to change every day, every year. If you're not in pursuit of that in your own life, then you're bored. You're starting to lose your sense of purpose. Our purpose, if anything, is to be curious and stay after it. And so I encourage you all, as we dive in here, to stay after it. The last point before diving into the science is around this concept of linear change. Linear change is not an option. This important new science, this revelatory new paradigm that the microbiome and the virome is at the center of human health, has come at the tipping point. It's come at this moment where we can see our own extinction event as a real possibility. When we see that we are in the midst, perhaps already halfway done with the 6th great extinction on Earth, we're not going to only see the demise of our species in such a scenario, we will also see the loss of some 90–95% of life on Earth. From the microbial to the macroflora and fauna, we are rushing towards an endpoint that will necessarily rebirth life on the other side of that extinction event, but it's right here. And so this critical discovery that the microbiome and the environment that we live in is actually the whole story of human health, our existence, our origin, and I believe our purpose is to come into a co-creative relationship with that ecosystem. But this new paradigm has come late in the game. It's in the 11th hour. We don't have time for hundreds of years to progress for the world to come to terms with the fact that the planet is not flat, or that the Earth is not the center of the universe. We don't have decades or centuries for scientists and physicians to start to ask the right questions around human health in the context of microbiology, in the context of virology. We don't have time for the next decades and centuries ahead for our politicians to finally catch up to this reality. And so what we need is an incredible revolution on a scale that could only be called metamorphosis. There is no possibility of linear change now. If we are going to survive, we need to change our mindset and our understanding of who we are, why we're here, where we're going, faster than any change we've ever made before. I would be overwhelmingly depressed about that, if not for the growing evidence that humans can change immediately. And if we learned anything from this pandemic, it is clearly that humanity, as 7.8 billion souls manifesting in those bodies, can actually act as one. And we can pivot on a dime. We can change our behaviors at the micro and macro level instantly. Many of those changes we made this year, we're going to find out were detrimental to the biology of the planet and ourselves. But we did show that we could do it quick. And so I want you to take away that realization of, man, when we all start to glimpse the future that we could create, we could get there mighty fast. And that gives me great hope and great excitement as to where we may be going. So where are we going? As we get this going here, I want to give you a sense here. I'm going to share the screen here because I want you to see this written down. This is an incredible piece of reality here that I want you to look at. Let's see here. I apologize. technical glitch there. All right, I think you're seeing this here. I'm gonna try it from this angle here. There it is. So I want you to take a look at this definition from the Oxford Dictionary, because I think this is such a critical place for us to start, and I believe this is where we're going to conclude too. But if we look at the Oxford Dictionary definition of nature, it's a noun, and it's defined as the phenomenon of the physical world collectively, including plants, animals, the landscape, and other features and products of the earth, as opposed to humans or human creations. I want you to just let that sink in. We defined nature as everything as opposed to humans or human creations. We literally, in our dictionary, have written ourselves out of the definition of nature, and we have put ourselves in opposition to it. This, I believe, this simple definition demonstrates to you how we came up with the collective narrative and the public health responses that we have seen in this last 10 months. What an astounding mistake we've made. And we're going to have to redefine our role within this nature, our participation in it, and the rest. If you haven't been part of our global health education since its origin, our first event was called the Virome. I desperately wanted to, towards the end of the year, I think it was October that we did our first event. I wanted all of you to start to get a grasp of what our virus is and what has cataclysmically changed about our perspective on these viruses. So I'm going to give you a quick primer on that 2 hour and 20-minute lecture that I did on the Virome. I encourage you to go back if you haven't seen that one, because I believe it's critical information for you and your family as you make some incredible decisions this year as to whether you're going to follow a common narrative towards your health paradigm or not. And so this data is critical, but in an effort to quickly give an overview to kind of frame the rest of this discussion today, I want you to understand that we now know that the virome is not at all limited in its genomics to something external to humanity. We now know that human viral DNA is largely where we come from. We have long thought that we just inherited all of our genes from mom and dad, and them from the humans before them, and them from the humans before them. But we now know through genomics that over 50% of the 20,000 human genes you inherited were inserted not by a human, but actually into the mammalian genome by viruses. And so we have, in fact, inherited, in fact, perhaps developed only because of the presence of viruses and their capacity to insert genomic information into mammalian DNA. We could not be mammals without these guys. Interestingly, at least 8% of your genes were inserted by RNA retroviruses. These have been made famous by the narrative of the terrifying HIV virus, the first retrovirus that was really studied in great detail. More money has been poured into that viral species than any or viral variant than any other in history, probably. But it turns out these retroviruses are in fact part of our origin story as well. These viral genes that have been inserted are critical for the ability for us to become mammals. Live birth is a very complex experience in biology. We went from the reptilian kind of experience of eggs and the aviary and the avian egg. And so it was birds and reptiles that suddenly gave way to this emergence of the mammals through the phenomenon of live birth. And making it capable of that, the mammal had to be updated with a retrovirus called ERI-15. This is a critical insertion that allows for the method for the DNA of mitochondria and the nuclear DNA within the sperm and the egg to achieve fertilization. And so human fertilization or any mammalian live birth fertilization could not happen with this important viral update to the genome of life on Earth. And so the retrovirus ERI-15 critical to allowing us to fertilize that first human cell. Deeper than that, once the cell is fertilized, the production of the placenta itself as a mechanism to sustain that life within the mother, again, a critical viral update. Hemo is the gene that is inserted by a critical RNA update there, allowing for the placenta to do its heavy work. As that life starts to emerge from the mother and go into the complex and dangerous reality of biology on planet Earth, whether it be the radiation from the sun or toxins from fungi or the environment that we breathe, whatever first insulted the human, has been important that we have a resiliency mechanism. And that RNA resiliency mechanism seems to be this HERV-H sequence that makes up about 2% of the total RNA found in a pluripotent stem cell. 2% of total genomics is an enormous number, because there's tens of thousands of genomic sequences within a single pluripotent stem cell. But to find a whole 2% of it dominated by this single gene is fascinating. And what's been discovered about HERV-H is it allows for that pluripotent stem cell to go past the bumper blocks that typically will keep a liver cell making a liver cell, or a kidney cell making a kidney cell. The things that keep differentiated cells going towards their own identity and reproducing only their own identity, the bumper blocks that are necessary for that can be removed or slid past by the HERV-H sequence. And so what it means is that we would have never had a stem cell if we'd not gotten this RNA update to the genome. And this RNA update allows for the transcription of the pluripotent stem cell such that it can slide across those differentiation blocks, bumper blocks, to produce a liver cell or a kidney cell or a brain cell, or et cetera, et cetera. And so that pluripotent capacity of stem cells and the regenerative capacity within the human, again, is the result of this extraordinary genomic update from an RNA virus. And so I wanted to give that overview quickly to remind you that we are from this origin of life. And viruses are thought primarily to come from bacteria, fungi historically, and then multicellular life, plants, and then ultimately the mammals and humans and the rest. But every life form is capable of producing the viruses. They're non-living life forms. They don't have a mechanism for producing fuel or eating or reproducing. They are simply genetic packages of information that update the genetics of the planet at a regular basis. And the sea of genetic information is vast. There is so much genetic information in the pre-human state of the world. Every liter of seawater contains about 10 to the 10 viruses. It means the whole ocean is around 1.2 times 10 to the 30 viruses. That's roughly 10 million times more than our stars in the entire universe, not galaxy, universe. 10 million times more than stars in the universe are the viruses within ocean water. Not surprisingly, it's not limited to the oceans. 1 gram of soil contains 10 to the 9 viruses, billions of viruses in a single gram of soil. Worldwide, interestingly, that soil content comes very close to the amount of viruses you'd find in ocean water. We're at 2.5 times 10 to the 31 estimates there. And not surprisingly, we find it in the air as well. There's some 10 to the 31 viruses in the air, and all of those are seeding the human body at 10 to the 15 viruses in a healthy adult human body. As I sit here and speak to you, I have 10 to the 15 viruses coursing through my body and bloodstream, cells, etc. And so not only is the world around me a sea of information, but actually the body itself is actually a haven for and the generative target for this viral sea of information. And that viral sea is interacting with my skin, my eyes, my ears, my nasal mucosa, my lungs, my gut. I am literally built to absorb this viral information and make a decision as to whether I'm going to integrate this new information, and always with the purpose of adaptation and biodiversification. That's really the purpose of the virome. Over and over again, it's seeking adaptation, meaning better and better response, gain of function response to the toxic environment, and diversification of capacity, biology, et cetera, diversification of the microbiology, diversification of the human genome itself so that we can survive in different environments. We can have resilience in some pockets of the planet where we need different adaptive responses than we need in other parts. So this sea of genetic information that's flowing through me right now is there for my own good. It's there for my updating. With that 10 to the 15 viruses in my bloodstream right now, it's important to recognize I have no fever, I have no signs of infection, I have actually no signs of overproduction of any of those 10 to the 15 viruses. In fact, I'm in perfect balance with this sea of genomic information in me. Some of this is being updated in single cells of my body. Some of it is being updated and putting genetic information into populations of cells in my body. In some cases, my whole body is capable of adapting to these viral genomics. In the cases of the ones that I gave you earlier, the ERI-15 and these ones that were necessary for mammalian life, not only did they get integrated into a few cells of some animal far back when, they got integrated into the nuclear DNA so that it would be passed on generation to generation. So right now, my body is determining what it's going to pass on to the next generation. Your body is in preparation. If you're in any sort of fertile stage of life, you're preparing that future generation through what genomic information you sequence and integrate into your body. And so I want to free up the fear of the fact that viruses are everywhere and they're in us and everywhere, and we know how to live in and around the virome. But I also want to just point to a really ridiculous old narrative we have around how viruses travel around the world. This past year, a big news launch in March, very excited, from Microsoft, launches an interactive map that tracks the global spread of coronavirus pandemics in real time to help fight the COVID-19 outbreak. What a relief to have technology jump in to show us how the virus was spreading. And as this map depicts, it kept showing these hot spots, and they would show us over and over again the airplane flying from China that flew over to northern Italy or to Germany or to Seattle or New York or wherever it is. And so we'd see these airplanes traveling along, and then boom, the map would show this spreading red dot. The purpose of this was to really, reinforce a belief that the travel of viruses, the whole theory of infection, is based on human-to-human transactions, human-to-human infections. When in fact, we've known for a very long time that viruses have been traveling the planet long before the first airplane. And so it's not surprising that we find that in this pandemic, like every other one, the viruses are traveling globally independent of human behavior. Now, early on in a movement of viruses, you can speed this process up, because for it to deposit the deposition rates of viruses as shown here in January 2018 of viruses and bacteria above the atmospheric boundary level, they're coding the whole world in this viral information. It actually takes weeks to months. And so those first couple of weeks of a shift of genomics or new genomic information coming out of Asia to reach the world, in those first couple of weeks, an airplane flight might slightly accelerate that into a pocket of community. But by the time that community has started to propagate it and has made variants of that virus, as we saw even in those first couple of weeks in Oregon, Seattle area, those first cases coming out in the United States, we already had 3 or 5 different variants of the SARS-CoV-2 virus by that time. And so we had these genomic variants. Meanwhile, by the time we had diagnosed those and genetically sequenced those, the aerosol spread, the aerial spread, not the respiratory droplet spread that you would see go 3 to 6 feet that might be passed on an airplane or something, instead going hundreds of miles, thousands of miles, and moving at a rate that's pretty startling around the planet. Within weeks of our discovery of viruses here in the United States, it was starting to coat the entire planet in this genomic information. And so we need to lose the belief that the virome is only passed human to human. It's passed through the air that we breathe, and it's independent of human breath that virus will move. That virus will largely move based on air currents and the particulate in that air. Evidence of airborne transmission of the SARS virus has been around a very long time. This was 2004 that this New England Journal of Medicine article proved this. So we've known for 16 years that these coronaviruses travel not through respiratory droplet, but are primarily put through the airborne method or the aerosol method of spread. SARS-CoV-2 RNA, this is April 15th, 2020, found on particulate matter in northern Italy and traveling irrespective there of human transmission. Identifying transmission as the dominant route of the spread of COVID-19 was concluded in May of 2020, and yet there was no change in public behavior at that point. At this point, we could have said, air travel is released. Air travel cannot compete with the global transmission that's already there due to air currents and carbon particulate in the atmosphere. And yet we continued to lock down harder and harder these air travel restrictions globally. And so even though we were scientifically making these conclusions by May, it would take, I think, years to get this into some sort of public health response that makes sense. If we again picture, those airplanes moving around and spreading, and humans being the main source of respiratory infection in the world, then we would have to ask, why the heck are we seeing all these hot spots in flyover states where hardly any air traffic occurs? A good example of it is this massive epicenter down here in the Four Corners area of Arizona and Colorado and New Mexico. Interesting how few airplanes land there, and yet such a huge explosion that's only really demonstrated equally there in New York and the like. And so we have to understand that there's ecologic terrain. There's terrain changes that are occurring to create all these hot spots throughout the Midwest. Look at all this stuff going on here in Louisiana, Mississippi, and the deep South here. All these hot spots are literally the flyover areas. These are places where no airplane lands. And so there's a relationship not to air travel, but to air pollution and soil, water, and air situations in these environments that are going to predict those outcomes much better than air travel. I want to kind of hammer this home with one other study that I find amazing, how many human viruses are circulating in the bloodstream of the general public and population, and is this a problem? This was whole genome sequencing data that was done on 8,200 healthy individuals living outside of Africa, which is important because of the conclusions they ended up making. These were individuals mostly in the US, Western Europe, and parts of the more wealthy areas of Asia. So these are actually the areas that are lowest in the endemic and pandemic presence of viruses like HIV, Hep C, and the like. And so 8,240 healthy individuals that have actually screened negative for the presence of these by standard methodologies of infectious disease screening. And they were found to have this population, 94 different known viruses in over 42% of the study participants. Now, they were only able to look for about 150 total viruses, because we actually, in that 10 to the 31, in those quadrillions and quadrillions, we can only isolate and identify a few of them at a time. And so we have a miserably small number that we can look for. But even by just looking for 120 common viruses, we find that 42% of the study participants have integrated that viral information into their genetics. And these included herpes viruses, anelloviruses, papillomaviruses, polyomaviruses, adenoviruses, HIV, HLTV, which are important in things like AIDS, hepatitis B, hepatitis C, parvovirus B19, and influenza. And in these 8,200 individuals, the amount of these previously thought life-threatening disease-causing viral SNPs were discovered. And so HIV was 5 times more prevalent than hepatitis C and influenza together. And so an extraordinary amount of HIV has been integrated into the human population. And this is, again, in a population that is thought to have very little HIV present. If this study population is representative there, we would have to conclude that there are actually 432 million people who have seen HIV and integrated that virus into their genetics. And that's as of 2016. In that same year, UNAIDS was estimating only 40 million, 37.9 million, less than 40 million people across the globe with clinical HIV or AIDS, which means that we missed more than 90% of the global population that have HIV integrated into their bodies. And so more than 90% of the population that harbors that virus may be completely asymptomatic for a lifetime, because they've taken that information in, they've come into balance with it, they've integrated the HIV into their genomics, and they are not producing any of the viral replication necessary to cause clinical HIV or AIDS. And so if 90% of people are asymptomatic with HIV, that would radically change our narrative around that disease process. And it could start to force us down an interesting pathway scientifically to say, what's unique about those 90% of people that are asymptomatic versus those less than 0.1% that will develop AIDS? And how do we move that population towards the asymptomatic version of viromic updates rather than viral infections? And so fascinating to think that we may be completely missing the boat on even the most feared viruses out there. HIV-1, if we start to think about the origin story that we were told, it kind of appeared in San Francisco in the early 80s and mapped back to Puerto Rico and parts of Africa. And so we had this origin story in the late 70s. That's now fallen apart completely. By 1998, we actually knew that story was incorrect because published February 5th, 1998 in Nature, one of the biggest publications out there, African HIV-1 sequence from 1959 was discovered in the blood bank in the US. And so we suddenly realized that the appearance of AIDS as a syndrome and then our subsequent isolation of the HIV virus in patients who have AIDS in the 1980s there was not an origin story. It was actually demonstrating a really radical change of terrain in the human body and our human relationship to a virus that had been around since the 50s at least, and maybe far, far beyond, earlier than that. But our blood bank only going back to 59, we're limited in our capacity to understand how far back it goes. But what's certainly clear is that something changed in our relationship to that virus rather than that virus leaping out of nowhere in the late 70s, early 80s. I like to look at hepatitis C as another demonstration of this radical change in our human relationship to the virome in the late 70s. And so again, remember our largest distribution of herbicides that were antimicrobial, and in fact, the tight junction injury, we'll cover glyphosate in a minute again, but the thing that opened up the innate immune system's first universal damage started around 1974. And that's, of course, when we see this massive shift in hepatitis C prevalence in the United States and in Western countries abroad. And so at the same time that we see HIV emerge and all of the HTLV viruses and the herpes viruses that have to be present for somebody to get AIDS, we had to change our relationship to all of those. Herpes, HTLV 1, 2, 3, et cetera, and the HIV itself, Hep C, all of these we changed our relationship to in the late 70s and early 80s. Interestingly, with just the realization that Hep C was prominent and prevalent within the United States, we made some really dramatic public health changes over the course of the late 1990s and early 2000s in the way in which we were screening for this, especially in blood banks and the like, and we seem to completely change our relationship to this virus again. And so fascinatingly, we have, without a vaccine, without any attack on this virus, we've seen the incidence of Hep C. In the orange line, you see estimated total new infections. In the blue line, you're seeing acute cases of hepatitis C presenting with fulminant liver injury and the like. And so both with acute presentation and kind of the chronic harbinger of the viral information within us, those incidence rates plummeted in the early 2000s as we got things really changed. So we fundamentally have shown that we can ignite our relationship to pandemics and we can get them to go away through a change of behavior. Unfortunately, we didn't look at it that way. We continued in this pharmaceutical model, this mindset and everything else, and that trend has gone away. This is now the number of acute Hep C cases since 2010. And so you can see that they've actually gone fourfold over this last decade. And so we have, over this last decade, fundamentally, again, changed our relationships to things like hepatitis C. How did the current pandemic begin? If we start to look away from Hep C and HIV as these kind of endemic diseases that have become so prevalent in the world around us, how did the current pandemic begin? Military or clandestine Chinese laboratories. That's become, one of the dominant stories out there, and many of my colleagues have poured an enormous amount of time to figuring this thing out, so whether this was the source or not. One thing that's interesting about this, just as kind of a aside in some ways, but of all of the efforts that scientists have tried to put new information out to change the common narrative over the last year, we have seen the most rigorous effort of suppression of voice, suppression of alternative perspectives than we've ever seen, I think, in scientific history. The level of censorship that we're seeing across the board is unbelievable as a scientist, that we're allowing this to happen, that we are comfortable somehow with the complete suppression of scientific pursuit, that active pursuit of understanding. We've discarded all of that to make sure we maintain this rigid narrative. And yet, despite all of that censorship, this story of this military or clandestine Chinese laboratory leaking this new virus out to the environment has not been suppressed at all. In fact, I think it's been in many ways kind of proliferated by both social media and media at large, as well as scientific communities, and has not been quashed. And so whoever's trying to control the current narrative don't seem terribly concerned about this piece of the puzzle. And there may be some reasons for that. It may have started as a gain of function viral experiment, but now released into a normal non-Petri dish environment, nature's going to have its way with that virus. And there's going to be a decrease in mortality in many variants, and then, excuse me, and there's the possibility for an increase in mortality with some of those variants. But those variants are in the hands of nature all of the time. Nature has been doing viromic adaptive gain of function behavior since its origin. That's the whole purpose of the virome, is to do gain of function in biology. And so these gain of function labs are new to the experiment, and they have new cool tools, CRISPR and things like this, that can edit the genome in ways that's perhaps never occurred before. But nature is always smarter than this. Nature's been doing this for 4 billion years just on this planet alone. And so 4 billion years of scientific experimentation of gain and function will never compete or be competed against by these military labs or otherwise. But the real thing that this serves the narrative well on is fear. And so if there's a consistent message that maybe that we need new technology because this is an unnatural virus, I think that's actually very attractive to the narrative that we need some new technological breakthrough of genomic, we need to genetically modify all humans now because there was this viral attack, this military attack. So I think it's convenient to the common narrative of we need technology if we believe the origin of this was technology. We've been told that 5G was maybe the origin of this, and a lot of my colleagues have focused a lot of attention that maybe the new advent of 5G radiation on large populations has caused this virus. But that would ignore the fact that coronavirus variants presenting almost identical to the very severe versions of COVID-19 that we saw this year occurred in 2002, in 2011 and 12 again. And so every 8 to 10 years, we're seeing a resurgence of these coronaviruses. 8 years is kind of remarkable how consistent that's been in the last 24 years. But we can map these coronaviruses back some 760 years in the human experience through some unique methodologies. And so hundreds of years of these coronaviruses and their adaptations, which will inevitably at times show more prevalence in the human population as it's needed. Remember, viruses are largely going to appear over and over again when new stressors occur, and those stressors then lead to the need for more adaptation. And so the adaptation is then expressed through the virome, and the virome is out there looking for adaptation mechanisms to reduce the toxicity. So could 5G or something like that, a new technology, participate in the stress levels of the organisms in and around those 5G towers, human or otherwise? Absolutely, could. But there's no way that it's the actual origin of this. And so I really believe that viruses have always been there, are always around us, are always in us, are always in some sort of relationship or failure of relationship within us. These stressors can create vulnerability. They can create a risk of morbidity and mortality in regards to our relationship to these viruses, but they're not the source of. And so that's where we can just kind of universally shake it off, everybody, shake off this fear of governments or this fear of 5G and all that. And let's come to realization that we're simply looking at a stress event within biology of the planet. And that stress event has caused changes in viruses, Hep C, HIV, and the rest. And those are bringing not an attack mechanism, but actually new adaptive behavior from our immune system. Viruses are forcing adaptation for an immune system to have a higher intelligence. And so when you shake off the fear and start to focus on the reality of biology as a single phenomenon, biology is not just human, it's all of life on Earth at once communicating through the virome. This seemingly complex slide here is simply demonstrating some of the most prominent viruses that have been pandemic since the 1970s. At the top, you have the avian influenza viruses in green. They jump species into the swine in the pink there, and the big jump occurred in the mid-1970s. which is again exactly when we started to see the emergence or change in our relationship to Hep C and HIV. And so we had some fundamental changes happening to our respiratory viruses in that same period of time. We start to see these species jumps more and more frequently. Interestingly, the swine are identical in genomics to the human. And this is new information that's just come out in the last 2 years or so. It's long been said that pigs were 98.5 or 99.7% identical to humans, depending on which papers you read. But a phenomenal study coming out of genomics lab in just the last 2 years did a beautiful job of the most complete genomic sequencing of the pig. And what they were able to show is that if they chop up the pig genome into 178 puzzle pieces and rearrange them, not changing a single base pair, but just shifting those 178 puzzle pieces around, they ended up with exact spelling of the human genome. And so the human genome is really identical in all of its matrix, just rearranged to the pig, which allows for a lot of biologic overlap when it comes to the sea of genetic information of the virome. And that's what you see happening here. You see from humans, the H3N2 seasonal human flu jumping into swine in the late 70s, early 80s there. And then from those swine, it can actually transmute into an H1N1 variant and then jump back into humans there. Which was first demonstrated in April 2009, but it was probably happening long before that. But it was the first time we had thought to take that travel through the species like that. And so whether it be from the avian to the human, which has now happened via the swine, or from the swine to the human, or from the human to the swine back to the human, the point being that our relationship to this genomic sea is always in movement. It's always in response to stress factors that are there. I find it interesting, this continuous pressure down towards the human through all of these avian and swine flus. If you look at the end of the graph by April 2019, all of it's pushing down into variants of H1N1, variants of human flu, which to me would suggest that we are the species most stressed now. We are the ones most vulnerable to an imbalance with our genomic information at a species level. We are pushing towards this huge imbalance vulnerability. This slide, we're going to come back to again later in the talk, is critical for your understanding of the way in which our NCHS, which is our one of our national public health organizations within the and working alongside the CDC, which is a non-governmental agency. It's important to know that CDC is actually not like the NIH and NCHS, which are governmental agencies. The CDC is a private agency that works alongside these governmental agencies. Nonetheless, this is reporting from the CDC website with the NCHS data showing the blue bars that make up the majority of this visual here. These thin blue bars that go vertical are the total number of respiratory deaths per week year on year here. And so these are kind of crunching that data into a 6-year or 5-year period here, 2015 to 2020. And you can see the seasonal rise in that bar as you see the waves coming through. You've got 5 dominant waves, and those are happening, of course, in the winter time of every single year. However, I want to show you how high those bars are between the waves. There's a lot of ocean blue there, and the waves are small ripples on top of a deep ocean of viral experience. The point here is that we have long understood that viruses don't suddenly pop out of nowhere in November, December and suddenly cause death. Viruses are in and around us every single day, and the rate of dysfunction of our bodies in relationships to that virome is very stable overall. You've got these small ripples of waves in the wintertime when our health declines or our environment declines, but it is always there. And so the blue lines are showing you that there's a very consistent death rate of about 50,000 deaths a week in the United States here, bar on bar there. And so very consistent distribution there. The green wave at the bottom is influenza. So these are the H1N1s and all of that I just showed you. So that tiny green waving line at the bottom is the percentage of all those blue bars that's caused by influenza. In other words, for the majority of the year, nearly 100% of the viral complications and death caused by respiratory dysfunction is not coming from influenza, and it's very stable over the course of the year. Influenza and ultimately pneumonia being the orange line on top of there pneumonia being the downstream complications of viral dysfunction within the mammal, that end stage of pneumonia where bacteria set into dysfunctional areas of fluid collection in the lung and the like, pneumonia accounts for a high percentage of the total deaths, but not all of it. And so even above that orange line is the percentages that don't end in pneumonia that still end in death. And so pneumonia accounts for a large part, but not all of the mortality. Influenza accounts for almost none of it. And so that red bar at the end of this graph shows the percentage of respiratory death attributed to COVID-19. Take a second to take that in. We said by April, May, June of this past year, we suddenly said that 100% of that blue sea of viruses of 10 to the 31, 10 to the 15, however many billions you want to think about, suddenly there was one virus that accounted for 100% of the annual patterns of respiratory disease. I would argue that is absolutely impossible. And I would also argue that we've never seen a vertical line like that in any pandemic. That vertical line is vertical not because of some new appearance of coronavirus or this new SARS-CoV-2 variant. That red line is purely an artifact of a new PCR testing mechanism that went global. And we suddenly discovered, like discovering HIV is in 427 million people and has been asymptomatic there, we started finding coronavirus signals in the bloodstream of people all over the world. And we suddenly said, well, this is massive pandemic happening. The pandemic was one of testing, and we took off with this whole narrative. Year on year, there's going to be shifts. We've seen, years where we have a terrible relationship with influenza and far more deaths from influenza than years previous to that. There's 2002 of coronavirus. There's 2012 of MERS. We've had these situations where coronavirus will account for a much higher percentage of this, but to suddenly say that we don't know what most of this blue line is to we 100% know that 100% of those blue lines is due to one virus among the 10 to the 31 that you're breathing today. It's just an extraordinary myopic story that we developed due to widespread screening with PCR, which is not a test of infectious capacity. It's the test of the presence of the virus in the environment in its interaction with our bodies. And so really bizarre that we as scientists were duped into this story so quickly. Is there a coronavirus? Yes, but can it possibly account for every single viral condition out there that's been stable for all of this time? There's just no way. How does the cell of the genomics of the antibodies present here? So I'm sorry, how does the cell produce a relationship that keeps us in relationship to this viral genomics if antibodies are not the primary mechanism of biologic balance? So this is pulled from another one of our presentations with the virome, but this is an overview slide that's showing us something called the innate immune system. If you missed the innate immune system event that we had 5 panelists on with PhDs and MDs talking about the innate immune system and all these different levels. Please go back and review that incredible 2-hour event that happened last month. And so this is a quick summary of that slide. The point being that the innate immune system is how we stay in relationship to viruses. The antibodies that we're trying to trigger with something like a vaccine are never the way in which we stay in relationship daily with viruses. In fact, you've already come to know this, that if you were to get coronavirus syndrome, that we would call COVID-19, and you lost your scent and went into respiratory distress and you were short of breath and maybe had some congestion, headaches and myalgias, fatigue and all that, if you had all of that happen, you had no antibodies present to that when your fever cleared, when your achy joints went away and all that. There was no, your body had not yet made a single antibody to that virus. In fact, the antibodies come on in a clinically significant way, some 3 to 6 weeks, if not longer, after the infection is already resolved. And so we know, even now as a layperson, that the antibodies are not the thing that made the virus go away in the bloodstream. The thing that made the virus go away in the bloodstream was a new homeostasis or balance with what we call the innate immune system. And the layers of the innate immune system are many, but the general ones are actually the microbiome, a diverse microbiology within our gut, on our nasal sinuses, within our eyes and ears. This microbiome interaction as our front line of relationship to the virome creates great balance, creates a great balanced mechanism. The bacteria can make antiviral compounds, the viruses can exert an antibacterial thing. The fungi can be antiviral, antibacterial, et cetera. And so microbiome has this huge checks and balances system that keeps us in relationship. After that, getting into the human level of defense is the tight junction barrier system of our gut lining, our nasal sinuses, again, the bloodstream at large, our blood-brain barrier, kidney tubules. All these boundary events are the tight junctions. And as those start to break apart under toxins in our environment, we start to become vulnerable and our innate relationship to these viruses shifts. Deeper to that then are these incredible enzymes that chop up genetic information as it flows into our cells. And so the 10 to the 15 viruses in my bloodstream right now are kept in check with my biology as I'm constantly chopping that stuff up with enzymes. A good example of these are the ApoBec3A and 3G enzymes that have been most studied in HIV. And HIV latency has been published on extensively, where they find people in Africa that have high amounts of HIV in the bloodstream, but they never go on to manifest any disease. And the thing that differentiates bloodstream presence of the ApoBec3A, I'm sorry, the bloodstream presence of HIV versus the syndrome of AIDS seems to be ApoBec3A levels. And so this retrovirology study pointed out at the bottom of this slide is demonstrating that. So this extraordinary, enzymatic scissors that are chopping up all this genomic information flowing into each cell, make sure that we only then go take that RNA, retroRNA, DNA from a virus and go either stick it into our genome within the nucleus or start to transcribe those proteins and make new viruses only under radical control of the genetic information flowing in through enzymes like ApoBecs. In addition, one layer deeper than that is an incredible system of control over the way in which we regulate and signal transduce for protein synthesis. And this is ribosomal control. This is an incredible demonstration in this study and many others now that signal transduction in ribosomal biogenesis is the most controlled biologic event we've ever witnessed now. The decision inside the plasma of a cell, inside that cytoplasm, the decision to take some genetic information, whether it be human or viral in source, to take that genetic template and then make a protein, make a complex enzyme, make a new virus, whatever it is the most regulated thing we've ever witnessed. Here it's demonstrating that there's 3 RNA polymerases that have to be in coordination for any of these to occur. And those are then regulated by 79 ribosomal proteins that have to be present in the right concentrations, in the right ratios to allow for RNA transcription to begin protein synthesis. And then there's additional 200 proteins that are things like helicases, splicing factors, chaperone proteins, non-coding RNA strands, and the microRNA and all the rest that also have to be present on those 79 ribosomal proteins for those 3 RNA polymerases to decide to make a single decision to replicate a protein from some RNA strand. What I'm trying to point to, I know this is complicated, I know this is confusing the hell out of many of you. The point being is that the decision to take genetic information, viral or human, is an extremely controlled step in biology. Lots of redundancy put in place so that we can stay in relationship to this sea of genetic information that we're breathing, touching, eating, etc. throughout our lifetimes. One thing that I would point out about this regulatory relationship, the innate immune system, I guess, before I move on, is infants. I think I ran by the number earlier in the presentation, but I'll bring it back up here because I think it's fascinating. Studies over the last decade have demonstrated that the stool in infants that are 7 days of age already have 10 to the 8th viruses per gram of stool. 10 to the 8th, and you're looking at hundreds of millions of viruses in every gram of stool in that infant. This is interesting because the infant does not actually have an adaptive immune system, which is the one that makes the antibodies. The adaptive immune system turns on between 3 and 6 months of age. So at 7 days of age, there's really no presence of a capacity for that baby to make antibodies to keep it in relationship with the sea of genetic information within its stool. Of course, there's viruses in its bloodstream and everything else, but to imagine that whole gut lining with this massive, almost 1 tennis court in surface area at that infant stage, already teeming with 10 to the 15 viruses per gram. There's billions and billions and billions of viruses held in perfect relationship with that infant. through this incredible signal transduction and the tight junctions and all these layers of the innate immune system. So I just cannot overemphasize the reality that we are put into balance with this. So how do we undermine this innate immune system? How do we create an opportunity for a pandemic to occur? Many of you have followed this over and over again, and I don't want to beat a dead horse, as they say here, but the glyphosate usage and cancer mortality in the United States is an interesting look at the innate immune system, a fundamental change in the innate immune system based on the spraying maps of glyphosate. Cancer is the end stage of a breakdown of the innate immune system's ability for regeneration, repair, and all this stuff. And so as we continue to challenge that space, we cause these high levels of cancer death. And if you take those spraying maps and overlap the Mississippi River tributaries, you end up with a demonstration of how we collect one of the most potent antibiotics and toxins for the tight junction barrier of the innate immune system. And we collect that into a single water system that ends in the last 90 miles between Baton Rouge and New Orleans, Louisiana. And on this map on the right there, you see this huge red spot of cancer and death all around that Mississippi, terminations, as it collects from this huge pink area in the United States, all the tributaries spraying of some 85%, 90% of the farmland in the United States, all coming into that one water system. An extraordinary demonstration of toxicity, playing itself out as a massive cancer epidemic that actually had never occurred in that part of the country. Here's a look at how that's done. This is our laboratory at Ion Biome again, showing in small intestine cells how small amounts of glyphosate, amounts that you would see in root vegetables grown conventionally, for example, blows the gut lining apart. So you go from this zipper together small intestine on the left, where those green tight junction lines are creating a coherent barrier, to the right, where the barrier's fallen apart and you have all these individual floating cells that are unattached to one another now. That's a demonstration of what's become proverbially known as leaky gut. Unfortunately, we've demonstrated the exact same phenomenon in the blood-brain barrier, in the kidney tubule barrier systems, and the like. Glyphosate is a direct toxin to this. And of course, we integrated this into the human experience in the late 1970s, right when we had these fundamental changes in the endemic and pandemic viruses that have become normal in our experience today. And so we've damaged this. And again, if not mentioned earlier, functions first and foremost as an antibiotic. So it kills the microbiome of your gut, the soil that it's poured on, the water systems that it filters through. It kills the microbiome, which again is the first line of the innate immune system, and then destroys the second layer of the innate immune system here at the level of the tight junction barrier systems. If we look at the global pesticide grids, this is demonstrating glyphosate applications across the world, and take a look at the hot spots of glyphosate that we have found. China and central Hubei province, where we saw this epidemic or pandemic begin, is among the most heavily sprayed glyphosate areas in the world. Glyphosate came off patent from Monsanto more than nearly a decade and a half ago now. And that chemical glyphosate is now made primarily in Chinese chemical companies. China sprays more of that chemical than anybody else in the world, sells more of that chemical than any other nation in the world. And so it's a massive toxic pool of this thing that undermines the innate immune system and our relationship to the virome. And so it's no wonder that we have viruses appearing in the human experience again and again coming out of Asia. Every year we go over to Asia and we try to guess which flu strains are going to show up in the United States based on the patterns of new H1N1 variants that are appearing in Asia as they undergo this intense biologic stress of the chemical agricultural system. Not surprisingly, northern Italy has the highest concentration in all of Europe there of glyphosate spraying. So Italy lights up as a whole, but if you look at a more specific map, you'll find out it's really northern Italy, but also other areas that we saw high rates of coronavirus mortality. We see again in those areas. Again, Midwest, the United States, big spraying, but also where we saw our first cases of SARS-CoV-2. Demonstrated in Seattle and area, Washington and Oregon, both with these high spraying areas. And so a potent demonstration of just how undermining the innate immune system can fundamentally change a global population. The other area where we use massive amounts of antibiotics that can nearly rival the 4.5 billion pounds of glyphosate is in the antibiotic usage in swine production. And so pork production On the planetary level, the United States is among the largest leaders. We have these massive toxic pools of pig stool out in North Carolina with Smithfield and these companies that produce an intensely disgusting amount of pork products under incredible antibiotic environments, which breeds incredible viromic and drug-resistant bacterial presence in those lakes of stool that are many millions of gallons, many hundreds of millions of gallons per lake. And so it's just a really toxic mess. And then you look at Asia, and of course, China out produces everybody else in the world with chemical production of swine. And so if you overlap swine production with glyphosate spraying agriculturally, you find out that, again, Hubei province is among the most toxic thing. But it's not the only mechanism through our soil and water systems that we break down that innate immune system. It's shown here is air. So soil, water, and air all contributing now. Not only does the air contain, in the United States now, some 75% of air samples taken are contaminated with glyphosate, but there's a deeper toxin that worries me here in the air, that is the carbon particulate from coal burning power plants, from the transportation industry and the like. And that is measured by a metric that we call PM 2.5, or particulate material 2.5, which is the microns of diameter of the carbon particulate in the air. And in this study here in the New England Journal of Medicine, we demonstrated in 2017 that every single air pollution and mortality in the Medicare population was linked. And then more recently in this study here around COVID, published in April 2020, we demonstrated with every 1 microgram per meter square of PM 2.5 increased, there was a 15% increase in the morbidity and mortality around COVID-19. And so that 15% increase with 1 microgram per meter cubed. And then you need to take a look at what's happened in the last couple years around PM 2.5 due to wildfires. And so it turns out that fires in Australia and then more recently in California can contribute not to an increase of 1 microgram per cubic meter, but many hundreds of micrograms per cubic meter. We saw levels as high as 500 micrograms per cubic meter in Sydney and Australia during those fires there. Again, California fires this past year, huge, tens and hundreds of fold increases in PM 2.5 in the environment just before we have what we call a coronavirus pandemic. And so we have seeded the air and the environment through agriculture and through air pollution with a perfect setup for a collapse of human health and a dismal demise of a stable relationship with coronaviruses and others throughout the world. Here's looking really specifically at PM 2.5 patterns in China here. Again, I wanted to underline that we can predict where these things are going to come from. It should never be a surprise that pandemics occur. We have the science and the knowledge and all of the metrics out there that are collecting this time in real time. You can actually go online and Google PM 2.5 New York or something like that, and you can in real time every day get highs and lows of PM 2.5 in all the major cities of the world. And so we are monitoring these environments, and we have the know-how and the scientific insight on how to predict where these things are going to come from. And this would give us huge abilities to go into prevention like never before. You want to prevent the next pandemic, you simply need to clean up the air in Asia, because that's where we see this intense thing. Change the agricultural system there, change the agricultural and swine production there, and then reduce the amount of carbon particulate in the air. Wuhan, right here in the center of this map, sits just south of Beijing, where we have the highest air pollutions, PM 2.5 levels from the energy sector and the like in the world. And they push down every time there's a northern high pressure front, will push this thing down and pull that right into Wuhan, where we see the highest levels of glyphosate and pork production and all of that. And so Hubei province right here is this perfect matrix for disaster. But it's not just location, it's also seasonal. And so the amount of carbon, in this case, CO2 and methane tracked here, but also carbon particulate in the energy production is going to be seasonal. At the top, you can see that there's very little toxicity of carbon by late summer and early fall. This is October that we're seeing clean there. Then as you go to the left of the circle here, by November 26th, which is really the beginning of the Northern Hemisphere flu season, as we call it, we see the carbon accumulating at an extreme rate in the northern hemisphere. At the bottom, you see April. So towards the end of the peaks that we see, April, May of flu season, look at the volume of carbon particulate in the air. It's an extreme amount of CO2, methane, carbon particulate that has accumulated in that northern hemisphere. Interestingly, you'll see that pattern dissipate as soon as the green foliage of spring and summer kicks in to pull that carbon back out of the atmosphere. So in the end, the seasonal waves that you see on top of the normal viromic baseline are going to map very carefully and very accurately to this amount of carbon in the atmosphere. So when we see a flu season or we see a high risk of pandemic spread, you can bet there's going to be high PM2.5. This is a quick look at that Australian fire situation. Many Australian cities and towns are struggling with horrible air quality during the bushfires. It was announced. Air quality has become the most toxic in recorded history there. Anything over 200, anything over 200 parts per million of that PM2.5 is considered extremely hazardous, and air quality index as high as 4,500 was seen. And so again, remember, every one microgram of per cubic meter of that PM2.5, 15% increased risk of an unhealthy relationship to coronavirus. And so when we see this amount of toxicity, it's an incredible likelihood that within the months of these fires that again happened right before the pandemic, right? And so in January 14, 2014, NASA has updates us that the stratosphere, the smoke's now reached the stratosphere and is affecting the atmospheric conditions globally. Smoke is expected to make at least one full circuit around the globe, NASA, US agencies. That was in January. By March, it become obvious that it was doing 2 cycles around the entire globe. And so we have coated the Earth in this PM2.5 in the months leading up to this event. And by January 2020, we already are making the announcement that this is a total setup for some sort of pandemic. Whatever virus is in the environment right now is going to have an increased penetration capacity into the human experience. And we have a high risk or vulnerability that didn't exist moments ago. Just as that started to settle down, the smoke levels in the stratosphere going away by June, July, we started to see coronavirus deaths plummeting all over the globe in June, July. Suddenly in August and moving all the way into late October of 2020, right when we should have had this big reprieve from any coronavirus scare, everything else, suddenly we had the worst fire hit season in history in California. That California fire season is the worst in recorded history, with over 4,257,000 acres burned in over 9,000 incidents, fires, individual fires, over 9,000 fires. And if you remember, many of these were thought to be arson events throughout the area. But whatever was causing these fires, it led to 4% of the entire state of California under flames. Massive, massive fire season that, of course, with the same air currents that you saw blowing across from Australia and Asia across the northern hemisphere, picks up now California and distributes that PM2.5 of that, 500 million acres burned is now in the atmosphere right from August to October of 2020, setting us up for yet another season of high carbon particulate in the air with high binding to viral content. Long-term exposure to PM2.5 lowers influenza virus resistance via downregulating pulmonary macrophage, which are part of the innate immune system, KDM6A, and mediates histones. So this study right here is showing that long-term exposure is to the PM2.5 not only will carry the virus through airborne particulate and everything else, it actually directly undermines the innate immune system while delivering the airborne materials of viruses and toxins to our bloodstream. And so these fires cannot be underestimated in their impact on the innate immune system, but not just fires, also refineries and being a huge source of these PM2.5, as well as coal burning and vents. And then in the developing world, really unforeseen, unfortunately, is the toxicity of kerosene heaters and cooking systems in the homes of developing nations. So Africa, South America being high parts of this, but also parts of Eastern Europe, parts of Asia, where there's still kerosene being burned in the home. Kerosene burning on these old systems leaves an enormous amount of PM2.5 immediately within the household environment. And so you can see what looks like a virus tearing through the family is actually a PM2.5 poisoning event. And it's being blamed then on the virus as kind of an innocent bystander as we do these toxic downloads of these carbon particulates. Not surprisingly, right before this pandemic, we see the highest fires in Australia. We see this toxicity mounting, and the EPA is scolded by Earth Justice that filed a bunch of lawsuits just before. This is May 2019, so right before the November, December kind of appearance of this coronavirus strain in Asia. Months before, Earth Justice sues the United States EPA for not alerting the public to some of the highest hydrogen cyanide levels that had ever been measured in these cities. Cyanide, it turns out, is a very potent toxin that, of course, causes vascular hypoxia and damage and death. Classically, it's one of the most famous toxins for causing poisoning deaths to humans. And that cyanide is again a byproduct of many refineries and other energy technologies out there. And we're not alerting our public that we have this very high amount of cyanide, which unfortunately binds to PM2.5. And so big fires with high levels of cyanide, we were a setup for respiratory disaster in 2020. Also binding to that PM2.5 is the SARS-CoV-2 virus itself. Now, this has been demonstrated for many years in influenza that binds to PM2.5, but this group in Italy went out of their way to go ahead and demonstrate that indeed that the SARS-CoV-2 RNA was found bound to the particulate matter of the PM2.5 in Italy at the time of their massive spike of coronavirus cases that were presenting as hypoxic injury. And so you've got SARS-CoV-2 binding to PM2.5, which is binding some of the highest cyanide levels ever. Remember, cyanide is also produced by wildfires, especially when you're burning infrastructure. So you burn houses and the like, and you saw how many houses were burned here. Jumping back, was 10,488 structures in California fire, for example. And those are full of formaldehydes and all kinds of insulation materials and plastics that when you burn, you get all kinds of hydrogen cyanide coming out of those materials as well. And so we were a perfect setup for events. So what is happening to these patients that are being poisoned like this on a global level? This, I want to take a pause on here because I really want to emphasize this part of it for you. I really believe that one of the biggest mistakes that we've made in the last year is that we categorized this event as if there was one thing happening. We said, oh my gosh, there's this new virus called SARS-CoV-2. It's a variant of coronavirus that's been around for 780 years, but this one is causing hypoxia, just like SARS did in 2002, just like MERS did in 2012. This one is causing this massive hypoxic injury that is leading to these high rates of mortality in New York hospitals, Northern Italy, Hubei province, whatever. And so we had this story that this virus caused this horrific syndrome of lack of oxygen, total system shutdown, multi-organ failure, and ultimately death in a high percentage of individuals. Unfortunately, we categorized then the virus as being responsible for that whole matrix. We didn't think about cyanide. We didn't think about PM2.5. We didn't think about the suppression of the innate immune system. We didn't do any of that. We just said this virus is responsible wholly for this syndrome that we saw called COVID-19, and this syndrome is fatal, potentially fatal in all kinds of people. That was the narrative we were told. When really what we should have recognized is that there's two things happening. There's actually those that are presenting with near-death or fatal experiences, and we can look to this JAMA article that looked at incredible study looking at 5,700 patients admitted to New York hospitals at the height of our pandemic mortality. And with these 5,700 patients, they published in this JAMA article all of the presenting symptoms, vital signs, and laboratory features to demonstrate that 5,700 patients developing this near fatal event, and we saw mortality in some of these ICUs as high as 88%, which is higher than any other viral syndrome that we've ever described before. 88% mortality. Ebola is around 30%. And so this would have been the most catastrophic, dangerous virus on the planet. that is coronavirus 19, if it was causing this. But if it was a virus causing an infection that was leading to these mortality events, we should see some signs of infection in this acute ill group that was dying from this hypoxic injury. But in fact, they were afebrile. The 5,700 patients presenting with this, intense near fatal or fatal syndrome, they were hypoxic with minimal respiratory changes, minimal congestion, and no chest X-ray findings of pneumonia or big fluid pockets in the lung. Very clear X-rays, yet they were hypoxic. They were not able to carry the oxygen in their red blood cells, and they were unable to deliver that. So they were developing total body hypoxic injury. Interestingly, just as they were afebrile, they also had no increase in white blood cell count, and they had no left shift in a white blood cell count that would suggest a viral infection. So there was no signs in these patients that were critically ill, these 5,700 patients showing up in New York hospitals, no signs of actual infection. They showed up with hypoxia, and they reported loss of taste, smell, and executive function. They couldn't think, they couldn't make decisions, they were horribly emotionally overwhelmed, they were bawling beyond any rational level of fear or anything else. They just were uncontrollably dumping kind of tears into this state. So very emotionally unstable, hypoxic, blue, sick group. The chest X-rays, both for SARS back in 2002 and the SARS-CoV-2 that we see now, is these clear chest X-rays. No pneumonia, no fluid filling this. Instead, there's on CT scan just done at the same time as the chest X-ray, you see a small amount of what we call ground glass, which is a fluid thickening around the airways, but much different than pneumonia and all of this. And this should not cause global hypoxia to the body. Notice that some 98% of the lung here has no pathology even visible within it. And yet they can't carry oxygen. And yet we kept thinking this is a virus causing some sort of respiratory syndrome where they can't breathe. And so we're putting them on ventilators. And oxygen, as it turns out, is very toxic to inflammatory tissue. And when you have inflammation starting and you pour high percentages of oxygen into that environment, you can actually make things much worse, which we absolutely did in these ICU settings as we intubated so many of these patients early on. Now, almost every hospital knows whatever you do, don't intubate a patient, get their hypoxia reversed through any other mechanism possible. And our outcomes are far better now. But it is interesting that we, again, attributed this hypoxic event to a virus when in fact, the problem was not a lung infection at all. It was in fact a change in the shape of hemoglobin leading to its inability to carry oxygen. This interference of oxygen carrying capacity is known as a very typical mechanism that's called histotoxic hypoxia. And I think I had another thing in here, maybe I missed a slide. That's all right, but histotoxic hypoxia is the syndrome in which cyanide is the most classic for. And so if you read about cyanide poisoning and you read the symptoms that are associated with cyanide poisoning, you're gonna see patients who have no signs of infections, they show up blue, they can't carry oxygen, no matter how much oxygen you put them on a ventilator, they can't carry it. Instead, to treat this histotoxic hypoxia, you just have to change the shape of the red blood cell. So anybody who's ever worked in a manufacturing environment, which are commonly use hydrogen cyanide, you have to carry your cyanide kit with you, which are three quick injections, sodium nitrate and the like. And these 3 quick injections change the shape of the hemoglobin molecule into something called methemoglobin, and it's suddenly able to carry oxygen again. So you can immediately reverse cyanide toxicity with a quick self-administered 3-punch shot system that we give to civilians all the time who are working in manufacturing settings. Unfortunately, we didn't look at this as a poisoning. We looked at this as an infection. We kept believing that these people that were dying of infection when I believe they were really clearly being overloaded with PM2.5 bound to cyanide that was being trafficked into the lung environment and ultimately into our bloodstream by the virus, because the virus is designed to actually enter the body through lung and vascular tissues and neural tissue, like our nasal sinuses, where we're seeing all this loss of taste and all of this loss of taste and scent in people exposed to this virus. It's because it's trafficking through the ACE2 receptors on the surfaces of all these tissues. And so the ACE2 receptor binds to the coronavirus and pulls cyanide straight into the cell to poison the human body with high amounts of air pollution that weren't being breathed, but were literally being smart targeted into the bloodstream by the innocent bystander of a virus that has been in our environment perhaps for a long time. So high cyanide, high PM2.5 with the coronavirus trafficking in, poisons the red blood cell, and we get 5,700 patients dying in New York hospitals of environmental toxicity, not of infection. So when I say what was happening this past year, I want us to recognize two things happened. We had many people exposed to a new SARS-CoV-2 variant of coronavirus. They typically got congestion, fever, intermittent sweats and flushing, malaise. Emotional overwhelm was very, very common with this syndrome. Uncontrollable crying, uncontrollable sense of impending doom, overwhelming sense of depression, sadness, grief, all these emotions pouring through. So this neuro kind of cognitive event with these fevers and everything else. That part is all of the inflammatory results that you would expect from the presence of a new variant of a virus that's triggering the innate immune system and ultimately the adaptive immune system to do what it should do to bring us back into balance with the virus. The separate group, which had the high mortality, were being poisoned, I believe, by the environment via the virus, but were not showing signs of infection at the time of their demise. And so we really should have seen very early on that there was actually two things happening at once. One was an environmental poisoning, the other was a new SARS-CoV-2 variant. And we would expect totally different outcomes and totally different management approaches for those two groups. Had we given cyanide treatment to all of those coming in with the 5,700 acutely ill, or in northern Italy, or anywhere where we had pockets of this high poisoning event, and we had treated them as cyanide poisoning, we would have had radically different outcomes. How come I'm so confident of that? Is because when we accidentally did start treating for this with hydroxychloroquine, we did see improved outcomes. And so hydroxychloroquine, which is that one that everybody loved laughing at our president for pointing out early on in February, March of last year, we were laughing at our president for recommending an anti-malarial agent to treat a virus. That doesn't make any sense if you think that the anti-malarial drug is just there to kill the protozoa of malaria. But it was accidentally discovered to treat the condition that we know as malaria. And what has long been known to do is treat conditions of poisoning of the red blood cell. And so it actually has abilities to treat versions of this methemoglobin need that you would get with cyanide poisoning. So you would get indirect treatment of cyanide poisoning with use of the compound there as the anti-malarial. And so wherever we've done that, especially when you put into play an anti-inflammatory regimen with that hemoglobin shift, you see an improved outcome. And so it's very exciting to realize that we already have pretty much proved that the environmental insults correlate perfectly with the occurrence of disease, and that the disease outcomes are bifurcated. One is a low-grade fever and all this congestion that has almost no mortality, and then a very high mortality event where the coronavirus is tagged PM2.5, cyanide, and other toxins in the environment, glyphosate and the like. And so we can poison the group and expect one outcome, or we can just see interaction with the innate and adaptive immune system of this new virus and see very low morbidity and mortality in that direction. So if we had done that bifurcated thing, we would have very quickly been able to say who's at high risk for the poisoning event, how are we going to manage that, who's at low risk for poisoning but at a high risk of an innate immune system imbalance, and how do we address that. So that's what I hope we do in the future, is something more along those lines. Interestingly, for as long as the agency has been there, the CDC always sends out a toxicologist along with infectious disease physicians and scientists to any new pandemic site. And so they've long recognized that the toxicity of the environment is a critical piece of pandemics, but they don't ever publish or talk about the findings of their toxicologists who are side by side with the infectious disease docs. They only will publish the information around the viruses to perpetuate perhaps our fear of those viruses. But the reality is they know and are measuring and have metriced these toxic environments long before I ever came along or anything else. And so CDC is tracking this information. They know this is a real part of pandemics. That's why they are sending the toxicologists out there all the time. We need to be alert to that. We need to start to demand that public information. We've paid for that information ultimately in some shape or form. We need to get that information and start to make intelligent clinical and public health decisions around those types of environmental decision making. This 5G thing has gotten a lot of attention. I do want to just point to the fact that we absolutely have nothing here. This theory is and blank. Like, we don't even know how to finish this sentence yet. Literally, I've seen, lots of small independent studies done at this point showing that 5G cell towers were in areas of high density population that had poor outcomes with it, but those are never done with multivariate studies. And if you're going to look at a high population of northern Italy and say that there's 5G towers there, and you say the 5G tower is responsible, then you have to do a multivariate analysis to make sure that the amount of PM2.5 and cyanide and glyphosate in the environment in northern Italy is not confounding that and creating a true and unrelated phenomenon. And so that's what we're really lacking is multivariate analysis, because of course it's in high population densities that we see 5G rolling out. And of course, that's where we're to see these other environmental toxicities that long predated the 5G network. And so is 5G safe? I would say there's an enormous amount of data that says absolutely not safe, but is it causing coronavirus? No, I think that's a jump we don't need to argue for to demonstrate the toxicity of 5G. Interestingly, there is one established mechanism of 5G that gives great concern around predisposition to respiratory death and making us even more vulnerable to cyanide toxicity and all that. And that's the phenomenon where they've demonstrated that 5G disrupts, both in plants and animals, the ability of oxygen carrying capacity of hemoglobin and other plant plastids and the like. And so disrupting oxygen carrying capacity within cells is a well-established downstream consequence of 5G exposure. And so we could certainly be, again, predisposing or making even more vulnerable to these toxic environmental poisonings through 5G radiation. So certainly profound amount of science suggesting that it's scary. We don't know what its full impact is going to be on humanity at this point, but we've become very accustomed to rolling out new technologies with absolutely no idea of the ramifications on our children, reproductive capacity, the planet, extinction, etc. We are so, as consumers, dialed into buying the next thing, whether it be a cell phone or a vaccine. We are the ones buying these things. We line up to extraordinary, patience to sit through lines for both of those things. And so we are, as consumers, pushing this envelope of technology before science and technology adoption globally before any scientific data comes out to suggest what we've done. So I'm concerned about 5G, but again, I want to free anybody up from thinking it's the source of the problem and it's another contributor. It's yet another layer of toxicity that we're exposing our children and our world to. This is taking a look at where I think we should have gone with PCR. So I don't want to go deep into the PCR thing because I think this has already been heavily trod and well educated on actually in the scientific environment, saying that PCR was never supposed to be a diagnostic kit. We should have never been trying to diagnose coronaviruses with PCR because PCR is far too sensitive. Just like those 420 million patients with HIV that's integrated into their DNA and are able to make those proteins, we should not be trying to detect these tiny little genetic sequences of RNA or DNA via PCR and then describe that as some sort of infectious process. The genetic information is not the infection. An infectious type syndrome that we see in the non-poisoned group and the other bifurcation, that's a demonstration of a dysfunction within the innate and adaptive immune systems where we get inflammatory cascades going out of control. And in those groups, we see the amount of coronavirus being a very poor marker of infection, but is an excellent marker of outcomes. And so if we, instead of using it as a diagnostic tool, had gone about using PCR as a prognostic tool, we could have actually used it to our benefit to say, here's somebody has actually very low viral content, but is showing up with hypoxia. This is probably a poisoning patient. And here's a group that is presenting without any hypoxia and has a very low viral load by PCR, and we can demonstrate very quickly that they have absolutely no risk of mortality. And then a very small segment has no signs of hypoxia, but has very high viral load, and then a cascading inflammatory syndrome that lasts weeks or months before they die of pneumonia and then respiratory failure. Those three categories could be differentiated using clinical presentation and PCR with quantitation. That's what we weren't told, and that's where it became such an ineffective and in fact grossly irresponsible use of a scientific tool, was when we did qualitative analysis by PCR, where we said, yes, you have it, or no, you don't. And if you have it, my God, you're at risk for all of that, instead of saying, you may or may not. It was this yes, no answer that we allowed the public to take information from. That should have been illegal immediately because it led to catastrophic mistakes in the ICUs that we were running, and it led to catastrophic mistakes at the public health and public health intervention, socioeconomic decisions, et cetera, because we thought it was yes or no. We thought this was a deadly virus that would behave the same in anyone and put everybody at the same risk, and then blah, blah, blah. We could have used PCR very effectively as a prognosticator. What is the viral load? What kind of replicates of that virus is in the bloodstream? And what are the clinical presentations? Could have completely changed the clinical and public health responses. So my argument is for quantitative rather than qualitative use of PCR. It's being done. They know how to use quantitative PCR. They know how to get that data to the physicians. It could have easily been the pathway. We didn't need new technology to do that, is the point. It could have been done. We didn't do it. We instead went for the yes/no category. So what is happening to these patients? Again, I just want to drive this home. Here's the histotoxic hypoxia verbiage here if you want to look that up. And I do encourage you to look this up, because you will read exactly what happened in these ICU patients showing up blue, and then ultimately their lungs filling up with fluid 2 to 3 days after presentation with no fever, et cetera. And then they start to get fever 4 or 5 days in. As the lungs fill with fluid, they start to develop secondary pneumonias, and then they die of pneumonia, not from the virus or anything else. And so cyanide as a histotoxic hypoxia event. Now, is cyanide the only poison that's there that could cause histotoxic hypoxia? Unfortunately, the answer is absolutely no. Glyphosate is another good example of a toxin that can bind to PM2.5 and travel at high concentrations in the bloodstream, especially when there's a virus to speed its adoption. But it's just the tip of the tip of the iceberg. If you look at nicotinamides and 2,4-D and all of these toxins that are used in the agricultural system, or you look at all the sulfur compounds that are common in the coal burning industry and the like, these things are all capable of inducing histotoxic hypoxia. So we can poison the world via viruses by tagging those viruses with PM2.5 and cyanide and all these other matrices of toxins to induce these injuries over and over again. And if we keep blaming the viruses, we will not take the right public health and ecologic interventions to mitigate these risks for future crises. Here again, the symptoms of histotoxic hypoxia: changes of color of skin, so you end up with blue all the way to cherry red in the most severe poisonings. Confusion, cough, fast heart rate, rapid breathing, shortness of breath, slow heart rate, flushing and sweating. This was the weird syndrome that all these patients were showing up with, and this does not look like a typical infection. And so our physicians should have been equipped with this, and we would have had a much different capacity to differentiate viral genomic instability and infection versus poisoning. Viral histotoxic hypoxia has been very well studied as well. So this is demonstrating that viruses, once the poisoning starts to occur, can actually participate in driving this. And this was actually most well studied in smallpox. And we actually used this protein in the smallpox vaccine that was in use until, was 1972, that we phased out smallpox vaccinations. But up until 72 there, everybody was getting this protein that actually can be seen as a critical part of this histotoxic hypoxia thing. So the virus and the viral particles that we were putting into the vaccines were already known to induce these histotoxic hypoxic events. Coronavirus typically does not show this as much as flu and all of that. And so of all of the respiratory things that we've got out there, corona is not the one that's got the high C16 prevalence there. And so again, I don't think it was the virus that's causing histotoxic hypoxia. I think it's much more a relatively benign virus that's carrying the environmental toxins into that environment. PM2.5, if you look into that literature, accounts for way more than respiratory death. You expect increased cardiovascular death, cancer death, all that increases over time with high PM2.5 levels. And across the world, this one, April 20th, just looking at just a couple weeks of data from March 16th to April 6th here in Spain, and looking down the list there. So just over a couple week period, looking at the number of excess deaths in these environments of high environmental toxicity, the ones that they could find by PCR, which again is far too sensitive to tell you who's really sick with that virus and who's just a carrier. But nonetheless, even with that, there was a large amount of excess death that wasn't being attributed to the virus itself. And so high PM2.5 would account to this pattern where we saw excess mortality in little pockets around the world that far exceeded just the respiratory diseases there. So PM2.5 would account for this broader total mortality event beyond the respiratory. So what starts to differentiate risk of the bad poisoning, hypoxia, histotoxic hypoxia, and death kind of pattern versus the low-grade congestion, fevers, loss of nasal scent, etc.? What was the differentiator between those two became really clear was these comorbidities. And so these are the leading comorbid conditions among coronavirus victims in New York: hypertension, diabetes, hyperlipidemia, coronary artery disease, renal disease. In most populations, they include in the coronary artery disease condition there also stroke, and so cerebral vascular as well as cardiovascular disease. So coronary and cerebral vascular disease, renal disease, which is also a form of vascular disease, hyperlipidemia, diabetes, hypertension. Interestingly, those comorbidities that are really accounting for the vast majority of it don't include the lung. And so if this was really a respiratory virus that was attacking the body, we would expect COPD and smoking and all these kind of lung conditions to be much more prevalent, history of pneumonia, these other comorbidities to be a more prevalent risk factor for this. But instead it was all vascular, which again is speaking to the entry point for the PM2.5 and cyanide through the ACE2 receptor that the virus binds to, which is in this vascular tissue, much more so than elsewhere in the body. Interestingly, all of those top morbidities and mortalities are treated with the same drugs, which I find just fascinating. So every one of those, hypertension, diabetes, hyperlipidemia, coronary disease, cerebral vascular disease, renal disease, all of these must be treated with an ACE inhibitor or a statin drug, or you're not practicing standard of care and you're at risk of a malpractice lawsuit. And so we are by law required to really put people on these by what we call malpractice and kind of standards of care. And so by kind of these unwritten rules of the game, we have to put these patients on these two drugs. And those two classes of drugs, it turns out, upregulate the ACE2 receptor. And so both the ACE inhibitor and statins upregulate the ACE2 receptor by which the coronavirus enters the body, pulling cyanide and PM2.5 into the bloodstream and the rest. And so we can tell you that the mechanism by which this poisoning is happening is in direct relationship to how much virus can you pull in. The carbon PM2.5 actually increases the clumping of those viruses. And so instead of having it aerosolized and distributed throughout the air in an even fashion, you get very high concentrations of SARS-CoV-2 around a single PM2.5 molecule. And then you upregulate the receptor for those clumps of virus with an ACE inhibitor and a statin drug. And now you've got the perfect poisoning delivery system for poisoning humans. I think this was unintentional. I think that we just don't have the foresight to think through these risk factors in real time, and we accidentally contribute to massive morbidity and mortality through our artificial perturbation of human biology. Why are we putting everybody on ACE inhibitors and statin drugs? These are screwing up some of the most fundamental biologic systems in our bodies, including the innate immune system and the anti-inflammatories made by the liver in regards to the statins and the lipids and all of that. And so we are destroying the biology that would keep us resilient to things like respiratory infections by putting people on these drugs. And so we don't do that risk benefit scenario. If we had, and we had gotten this news late in December that, oh my God, there's this new Chinese variant of SARS-CoV-2 and it's going to be 30 times more deadly than the flu, which is what we were told by many of the regulatory groups, we should have immediately started making public health decisions of like, oh my gosh, coronavirus enters through ACE2s. We better get everybody off statins and ACE inhibitors in the United States immediately. Within a couple of weeks of stopping ACE inhibitor and statin drugs, the ACE2 receptors will downregulate again and back to their normal thing. So we could have decreased the risk of the population if we had made a really aggressive change. The risk of increasing cardiovascular disease and stroke by stopping those two drugs takes years to measure the consequences. There would have been no measurable public health consequence to stopping ACE inhibitors and statin drugs over a short period of 12 months or whatever needed to let this coronavirus things pass. But we didn't do that kind of proactive change. Instead, we kept these patients artificially, chemically adapted to increase their absorption of this virus and whatever it's carrying with it. Similarly, we knew very early on in this epidemic that this one was just published in March 2021, but we knew even as early as China, but also reinforced in northern Italy, that vitamin D deficiency was a huge potent contributor to morbidity and mortality for this virus. And this is not surprising because those little waves that I showed you in the seasonal variations of respiratory infection correlate with our months in the Northern Hemisphere where we don't get enough sunshine, and we're then vulnerable to a higher rate of imbalance with the biome at large in those months where we're vitamin D deficient. There's been a remarkable amount of effort and demonstration of how vitamin D interacts with the innate immune system and, in fact, the adaptive immune system to improve our balance with the virome. I'm not going to go through all of those studies, but I just thought this one was really pertinent here. This study showed that 59% of patients hospitalized with coronavirus in COVID-19 were vitamin D deficient with levels less than 20 on admission. Ideal in my clinic, we like to keep people up in the 50 to 80 nanograms per milliliter in our clinic. So a really significant deficiency in regards to the biologic consequences there. And then they found 79% of the critically ill admissions were vitamin D deficient. And so the vitamin D deficiency on admission was associated with COVID-19 mortality with an odds ratio of 3.87. So that's almost a 4-fold increase of mortality in patients with vitamin D deficiency versus those that were vitamin D replete or had enough vitamin D. Interestingly, those vitamin D levels were independent of other risk factors like age, chronic disease, comorbidities, coronary disease, hypertension, diabetes, and the like. And so they did the multivariate analysis in the study to show that as an independent risk factor, vitamin D deficiency increased mortality by 4-fold. If in fact we lost the 400,000 patients in the United States from this condition, as we've been told, imagine what a 4-fold decrease would look like if we would simply said at the very beginning of this, we have a seasonal virus coming, it's a coronavirus, it's already showing some high mortality. We're going to decrease ACE inhibitor and statin use throughout the United States population, and we're going to put everybody on vitamin D. Everybody needs to be on 5,000 IU vitamin D for the next 3 months. We're going to load everybody's reservoirs up, and we're going to 4-fold decrease the amount of mortality and morbidity from coronavirus. We could have done that. We knew it. We still know it today. And yet, is there anybody coming out universally recommending that everybody get on some vitamin D before the next seasonal respiratory season? It just is not happening. Something else that we did to undermine the innate immune system that I think is profound and will take us years to really account for fully, and I think it was devastating, is the contribution of fear. To fear or not to fear, and what are we going to fear? We feared this virus because we thought that it was responsible for all of these poisoning type events, these horrible, bizarre deaths of respiratory failure and everything else. And we got this message that by PCR, some huge percentage of the population has cases. And so all these cases, and every one of them has this high risk of death, blah, blah, blah. We created this huge fear narrative. This study is an old one, looking at 2002 to 2011 mortality rates of people with anxiety disorders. Anxiety is acute stress reaction, which is the most mild form of generalized anxiety disorder, shows a odds ratio for mortality at 1.69 of all-cause death. 1.69 suggests a 69% increase over the control population that does not have acute stress reaction. So 69% increase in all-cause death. Not surprisingly, it's almost a 3-fold increase in death from unnatural causes, which would include things like suicide, substance abuse, overdose, etc. And then natural causes, still a 30% increase in death over the population. And then you go down the list: panic disorder, generalized anxiety disorder. That one's a big standout. Generalized anxiety disorder with, again, 62% increase all-cause mortality, nearly 2.5-fold increase in unnatural causes, etc. So looking down that list, all of these phobias and fears and inductions of fear increase mortality over and over again. And so with this narrative of there's this one virus that's causing these horrific deaths, and not giving the context of the event to understand that we were going to have completely different risk factors depending on our age, our comorbidities, the drugs we're on, blah, blah, blah. We just had this one paradigm that we are pushing, was that there's a terrible new pandemic upon us that could annihilate you and your children, or your grandparents being a more common part of that narrative. This is getting at what I have found in my clinic. All of the coronavirus patients that we've had, both in our clinic and in our extended kind of social communities and the rest, this has been the one thing that I've zeroed in on again and again. I now think it's the #1 modifiable risk factor for getting coronavirus, which is sleep. This happens to be just looking at one version of sleep disorder, which is obstructive sleep apnea. But this was looked at as, again, in a multivariate analysis, and it showed 8-fold greater risk for COVID-19 infection compared to age-matched controls and comorbid adjustments. And so adjustments for diabetes, obesity, and other conditions that are common with sleep apnea patients were adjusted for, and still they show this massive 8-fold increase in risk of COVID-19 with a lack of good quality sleep. And what I find in my clinic is that insomnia is actually even more common than obstructive sleep apnea. And over again, especially the healthy young patients that ended up getting this condition were notoriously getting poor sleep hygiene in the weeks and months and days, and literally like the 5 days before. If you even look down at the week before symptoms develop, sleep deficiency was a dominant and perhaps unifying feature of those younger, healthier individuals. And you're like, how did this person get corona? They don't have hypertension, they're not on ACE inhibitor or ARB or statin. What causes, and they're not in a big inner city environment. What sleep is I think one of the most overlooked secret toxins here to the innate immune system. That's why as soon as we did the virome, our next Global Health Education Summit was on sleep. So if you haven't seen the Sleep Global Health Education Summit, go back and see that one. We are being very intentional, change human biology, change the planet's risk of extinction through these events. So if you've missed some, go back, they're important. They're gonna change the way you think about your health, the way you think about life. And so please go back and see the sleep study, because I think it's, or the sleep section of Global Health Education. I think it's incredibly important information. If you don't know how to sleep well, you are at high risk, 8-fold increase. Again, think about if we had modified the environment, knowing that there was some pandemic coming. What if we had said, look, most important things, you need to fix your sleep hygiene, get on vitamin D, go through these steps, get off your statin drugs, et cetera, et cetera. 8-fold decrease if we had just fixed sleep apnea. Oh my gosh, get screened for sleep apnea. Or if you have sleep apnea and you're not using your CPAP machine, there's a coronavirus coming. It's going to be an 8-fold reduction in your risk of getting that virus if you get the sleep. So put on your CPAP machine and get the sleep you need. Higher per capita GDP, interesting socioeconomic factors that led to increased morbidity and mortality from COVID-19. The higher per capita GDP correlate with increased obesity, and obesity is a marker of kind of excess calories in a system, and COVID-19 mortality. And so obesity and GDP mapping very closely with COVID-19 mortality. Interestingly, this study also shows, and this study was done in Canada, so the Canadian health system has a much better record keeping than the United States does because of our fractured healthcare system. But in Canada, they demonstrated this amazing finding that increased testing correlated with increased mortality from COVID-19. And so countries with the highest rates of COVID-19 testing by PCR show the highest mortality from this virus. And so all of the things we were doing with contact tracing and trying to find out who you've been exposed to, and then everybody's been exposed, that should get a test. Every time we responded that way, we increased mortality. Why would that be? Why would testing increase mortality? We see this again and again across many different disease types in Western medicine. When you see an increase in mortality with increased testing, it means that our medical response to a positive test is toxic in and of itself. And so this is measuring the mistakes made in the medical care system in response to testing information. And so this has been done again many times in the past. It's not surprising that we find this pattern again with COVID-19. Over-testing leads to over-medicalization, leads to bad decision-making and poisoning ultimately through our pharmaceutical methodologies that I am such a part of in my past history as a physician. Alteration in natural immunity for coronavirus and other respiratory viruses. This is very inflammatory information at the socioeconomic level, if not at the biologic level. So alteration in natural immunity for coronavirus and other respiratory viruses has been demonstrated manyfold. This study looking in 2012 showed a 4.4 increased risk of non-influenza respiratory viruses in children receiving the flu vaccine. Let me repeat that or rephrase that. In children who receive the flu vaccine versus those that do not, there's a 4-fold increase in non-flu respiratory viruses. In 2014, Japanese study confirmed that this through large-scale public health modeling. So the 2012 study was a relatively small study with a few thousand individuals, but then in 2014, it was confirmed through very large-scale public testing of hundreds of thousands of individuals to demonstrate this increased risk. it would be one thing if we just knew that as a theoretical risk, but unfortunately in 2017 and 2018, it became knowledge, and this became specific, unfortunately, to coronavirus. So in this study from the Department of Defense, we saw that influenza vaccine status in the Department of Defense personnel increased risk of coronavirus infection specifically. And that was as much as a 4-fold to 6-fold increase in coronavirus the following year. So once again, if we were going to be prospective and preventative mindset for a coronavirus pandemic, SARS-CoV-2 discovered in December of 2019 in China, and this is now threatening us, and we have a 30-fold increase over flu we're afraid of. Well, we should have certainly, if we thought it was 30-fold times more dangerous than flu, immediately stopped all influenza vaccines as of January of 2020. We did not stop it. In fact, we increased, and to this day, continue to increase the public health awareness of the need for flu vaccines. Right now, you watch TV, you are going to see more and more, and have already seen more and more commercials for flu vaccination. In the setting of one of the largest coronavirus pandemics we've ever had, we are pushing flu vaccines against the science that's telling us that everybody who is immunized, and it was children in 2012, it was the whole population of Japan 2014 age groups, and then in young healthy individuals in the military, the Department of Defense study in 2017-18. So across all demographics, we have shown that flu vaccination increases the risk of coronavirus, and yet we keep doing it. And so we have to stop believing that the viruses are attacking us, and we have to stop coming to some sort of paradigm where we trust that our exposure to the virome at large is the way in which we interact more intelligently with that virome tomorrow. The way in which we interact with all of nature today is the way in which we interact more intelligently tomorrow, okay? If we don't come to this realization, we will continue to make these catastrophic mistakes. Influenza vaccination increases the odds ratio for coronavirus by 36%. This is, again, in that narrow study of healthy young individuals in the military. So really frustrating that we are falling behind on this. Already covered this, the fear is an independent risk factor for death. The new SARS pandemic. I want to take a look now just kind of at the beginning of this whole thing and where we could have made some public health decisions, not because I think anybody did anything completely wrong and was intentional about it. What you see is a series of mistakes made that are fundamentally made all of the time in medicine. So if this was an isolated event, I could say maybe there's a conspiracy theory, maybe somebody was really trying to screw up the public narrative, but I have seen the same mistakes made at the public health statistics level over and over again. In my clinic and other clinics, we make these mistakes of applying population statistics to individuals, and we do all kinds of messed up things with statistics to confuse ourselves. But we certainly did this in these early parts. There was a cruise ship in Japan that you might remember that came into Japan on February 4th. They were the first to start to be concerned that they had a coronavirus outbreak on this ship. The ship tested all 3,700 passengers and crew and discovered 712 cases there. And ultimately, there was 122 deaths, which shows a mortality of 16%. That is one of the highest rates of native virus causing death in a population in decades. And so this was scary. If these numbers held up and these numbers were correct, and that this very first PCR test that was out there was all accurate, we showed an incredible high mortality with this thing. A deeper question should have immediately been, how did 122 patients die on this cruise ship so quickly? What other factors were there? What was the toxicology of that ship? Because simultaneously to that ship coming in, 2 other cruise ships landed elsewhere in Asia, February 6th through March 7th, with 54 were those excursions. And they had a total of 5,400 people on those ships. And of those 5,400, hundreds were sickened with something that is similar to what we would call the SARS-CoV-2 experience. They had the respiratory issues, some of them were some hypoxia, all of this. But only 12 cases were PCR positive among those tested, and they were only testing sick individuals. They didn't screen the whole ship like the Japanese study had done. But what they saw, relatively low amount of PCR positive things. What this is, one of the things it's just showing is how poor our PCR was. We had a very high false positive rate, very high false negative rate in these early iterations of these tests that were being put out as far back as February of 2020. And so Interestingly, of all those sick individuals, with 5,400 patients on these cruise ships in tight quarters, blah, 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 there was no deaths, and in fact, no hospitalizations from those groups. So there was radical differentiation between a really high mortality level at 16% versus no mortality in even a larger population that were screening positive for the same virus, perhaps at lower variants. And so we were seeing high population variability in that time with these early testing kits and really wondering what was going on. I think that somebody needs to go back in time now and figure out why we saw 122 deaths, because nowhere have we seen this type of mortality now play out with SARS-CoV-2. And so there was a toxicity event on that boat that happened. It could have been a gas leak. Carbon monoxide poisoning would have been a very common thing on a big ship like that. And again, new pandemic, everything else, it gets covered up as, oh, it's a coronavirus event, and nothing else is responsible. And so we should go back and look at that event because it's a standout. There's not been any repeat of anything like that on any cruise ship or otherwise. In the United States, our first experience with the cruise ship environment was the Grand Princess, which you may remember landed in Oakland on March 9th and was quarantined there for another 14 days. It had already been out at sea. The Grand Princess had been exposed to SARS-CoV-2 with one elderly patient that had become severely ill down in early part of the cruise as it had left California and went down to Mexico. That patient ultimately offloaded to the hospital in Mexico. And then the cruise continued towards Hawaii, as I recall, and then ultimately was called back to Oakland before it landed. But it had been out at sea for weeks, and all of the right conditions were there to have a catastrophic event go on. The PCR was flown in by helicopter after it had been in bay for already a week or so, as I recall. And so it had already been quite a while into the progression of the syndrome on the ship there. A couple hundred people had reported some symptoms. The kits were limited to 1,100. So even though there was 3,700 passengers in the crew, only 1,100 kits were delivered. And so they screened a bunch of crew, including those that were symptomatic, and they found 100 individuals that were positive for PCR. So what they found in that was actually a clear incidence rate or prevalence of the virus in the crew. So 1,100 people, they found 100. So just under 10% of those tested were positive, which then could be extrapolated to the whole group. But instead of that, we see this classic statistics mistake being made where they said, well, it was 100 of 1,100 people. So that's a 3% mortality rate or morbidity rate, because ultimately there was 3 deaths that were reported weeks after the people were pulled off the ship. Nobody died while they were on the ship, which is telling. Again, increased testing increases mortality. So you screen for 100 positive, you then take those 100 positive off the ship after quarantine and you put them into hospital settings. Nobody had to be evacuated from the ship, which again, is telling. They were actually relatively healthy, which is pretty amazing because this should have been the sickest of the sick. Average age over 70, this should have been a disaster. And yeah, everybody, even symptomatic, were hanging in there on the ship. And then we get them off and weeks later, three die. So then we were told that 3 deaths out of 100 is a 3% mortality rate, which is, was told to be 30 times more deadly than influenza with a 0.1% mortality rate. And so we developed a narrative from the Grand Princess that this thing was 30 times more deadly than flu. However, we didn't do the real prevalence. We based it on this 100 cases and that were positive for PCR, forgetting that we had not tested the whole ship. So if we had extrapolated that and tested the entire ship, we would have expected of the 3,700 passengers to see 330 cases of the condition, because we had already determined that incidence. And so 330 cases among the 3,700 crew and 3 deaths, we would actually see a 0.9% mortality instead of a 3% mortality. So still high, still scary. But that's like in the league of the 2017 flu, 0.7 to 0.9% mortality with that flu. And so we were quickly getting down into flu-like, bad flu type numbers once we did the statistics right. But then we did something further disastrous, which this ship with this highest risk population is a terrible way to have a case study to extrapolate to the population. And so average age over 70, confined to tight quarters for over 3 weeks, and we only have a 0.9% mortality rate, we then extrapolated that to the whole public, saying, oh my gosh, we could have a 3% or 0.9% mortality with this virus globally, when in fact we know that the vast majority of the population can have a radically lower risk than those 70 and above people on the cruise ship held within close confines, all of the wrong things going on. There was no reason we would have expected the 0.9% mortality rate to hold up with at all any length of experience in the general public outside of a cruise ship with elderly people. And so we make these broad public health statements. Here's something that's 30 times more dangerous, or 9 times more dangerous than the typical flu, whatever it is. We are making these catastrophic mistakes at the science level, and then ultimately at the public health decision-making level, based on these case studies of the highest risk individuals. All right, I think I already covered all this. I'm not going to reiterate that. This is just demonstrating the way in which the public health groups like to cherry pick data, and they do this subconsciously. I don't think this is even intentional. But at the same time, this is February 28th, 2020. At the same time, we have all these cruise ships all over the world showing up with SARS patients, SARS-CoV-2. This ship coming into the port of Maryland in Baltimore has 82 sickened people. But none of this is now that no public health officials showed up to comment on this. So this is literally the PR individuals, the public relations spokeswoman from the Royal Caribbean that's making these statements. No public health official weighs in on this situation. But the public health spokesman, or the public relations spokeswoman, says none of the passengers had transited the COVID-19's regions of impact or interacted with anyone in those regions for 14 days. Therefore, we do not consider these coronavirus cases. An elevated number of guests and crew had flu-like illness, and some tested positive for influenza A or B and were placed in isolation according to medical protocol, she said. The CDC did not request coronavirus tests for any of the cases. The CDC did not respond to a request for comment from the journalist. And so because it wasn't fitting the Microsoft map of somebody flew there and there was Hubei province and all of this theory as to how viruses spread and all of this, because it wasn't fitting that map, they didn't look at this cruise ship to find out what was going on. And I guarantee you we would have found some PCR positive people within that group, because it was matching the patterns that months later became obvious were global. But because of our mindset, again, of the way in which viruses spread and our respiratory droplet protocols and everything else, we can get so myopic as scientists and as public health officials that we miss these opportunities to better understand pandemics in their early phases. What did we learn from MERS coronavirus? I think that instead of going in depth into that, I want to just point out that epicenters can occur all over the world. It doesn't have to be, South Asia that all these viruses are coming out of. MERS developed in the Middle East out of a toxic event that occurred there likely. So when we start to see these patterns of here's a new pandemic coming from some new region, we should immediately have our radar up of what's happened in that environment. Was there an oil spill? Was there some new toxic exposure due to change in manufacturing of electronics or something else going on there? We need to start being alert to when we see changes of patterns of pandemic or disease occurrence. We need to stop blaming the virome and start looking at human behavior to understand these natices of condition. As I've said before, this does not show up as pneumonia. It shows up as this ground glass injury in small parts of the lung when you see the hypoxic injury. And then weeks later, the patient dies of acute respiratory distress syndrome, or ARDS, in the ICUs, which is not related to any virus. This is the immune system's dysregulation that leads to ARDS. And so again and again, we've said that 40,000 or 400,000 people have died from coronavirus. We would be very hard pressed to demonstrate that anybody has died directly from this virus, because the virus, again, creates a relatively mild syndrome. What creates the vicious syndromes are the poisoning events, which are very unique and hallmarked, and the downstream consequences of an unregulated or dysfunctional or imbalanced immune system that leads to these systemic inflammatory events downstream weeks after exposure to something like coronavirus or flu. Flu does the same thing. Any virus is capable of starting the immune system into an appropriate response. If that immune system does not have the coping mechanisms and all of the regulatory stops and checks and balances and whistles and everything else, it will tumble into these severe inflammatory conditions that are almost uniformly fatal. And we certainly saw that going on in those ICUs. When we hit that 88% mortality in a few of those ICUs, that paper was ultimately withdrawn because they diluted it out with a bunch of other hospitals. But the reality was we did see mortality rates in these grotesque numbers in a number of ICUs at the height of March, April timeframe in New York and other cities around the world. So we know why we were doing that injury now. We were treating them as if they were pneumonia patients or had respiratory fluid in their lungs, when in fact they didn't have fluid in their lungs yet. That would only show up days or weeks later. And so we were mistreating these patients over and over again to again, our misperception that this is a virus instead of the possibility that there were other mitigating circumstances of toxins that were contributing to their symptoms before they were put on the ventilators. I think I've pounded this earth justice stuff into the ground here and everything else. Just a quick review of the cyanide stuff and everything else is available here in the literature if you want to dive deeper on that. This one looking at the treatment paradigm and how effective it is, especially when administered in hospital. So a great look at the treatment of cyanide poisoning if we had gone down this avenue instead of the kind of antimicrobial and respiratory failure kind of model. I think we could have, again, seen that much different scenario. Pre-existing conditions, I don't want to go too much further into that, but I just want to provide more literature here for the ACE inhibitor angiotensin contributors to this increase. This is March 23rd, 2020. This publication came out. And so again, very, very early in this epidemic, we knew the data we could have modified, especially for these ICU patients, and take them all off statins and decrease whatever you can do. Anybody who's a family member of somebody who's become sick, get them off of the ACE inhibitors and statin drugs. Clean up that pharmaceutical environment around these patients to help them self-regulate back into a relationship with the virome. Age is an independent risk factor. We saw this in the ICUs as much as we saw it anywhere else, but it's obvious. It's seen throughout all history of respiratory viruses that the older you are, the more likely you are to die from respiratory causes. And the rates of respiratory death are going up around the world, and they have been for decades. So this is looking at the Hong Kong respiratory mortality. And in Hong Kong, you can see over the course of 1981 to 2017 here, this consistent rise. There was a big spike 1995 to 1998 there, and then a steady rise since then over the years. And that mortality looks like, oh my gosh, we have more and more respiratory death. We must have worse and worse viruses in Hong Kong every single year to be accounting for all this horrible respiratory death. The blue bars are males and the pink bars are females here. Both across genders, you see this climb in cases and number of deaths, accrued death rates from pneumonia from Hong Kong. So what the heck is going on in Hong Kong? Must be a terrible pandemic going on there. No. As soon as you adjust that for age, you see that year on year over the same period of time, other than this toxic event that happened between 95 and 98, there's been a very slow, if any, steady increase in the rates of pneumonia and when age adjusted. What this demonstrates when you go from this to this is that this whole pattern of mortality is not from some new virus, but is because there's an aging population. So there's a higher and higher percentage of people between 1981 and 2017 of age greater than 70 in Hong Kong. Because of their aging population, more of them are going to die every year from respiratory cause, and it has absolutely nothing to do with new viruses or new attacks. It has everything to do with the aging population. So China and Hong Kong do a good job of keeping these statistics age-related and population-adjusted. We don't tend to do either of those in the United States. So all these cases of COVID and coronavirus, we do not age or population adjust those numbers because they look too benign otherwise, perhaps. And so we tend to overstate things by this. Other thing I want to point out when we're to put all these public health statistics in context is that if we look on the left at China or on the right in northern Italy, COVID-19, February, March, at the very height of their mortality rates from this event, COVID-19 accounted for a very small amount of total death in those countries. Not surprisingly, stroke, ischemic heart disease, lung disease were the top in China, and more typical of the West is ischemic heart disease, dementia, stroke, and lung cancer in northern Italy. And so you see a difference between East and West morbidity and mortalities, but in both cases, COVID-19 is a tiny amount of the total death happening in those populations at the height of the pandemic. And so we made these extraordinary socioeconomic decisions, social isolation, economic collapse, all of those we made around a very small percentage of total mortality. We should never make gross economic decisions, and we should never make, gross biologic, fear narratives around viruses. They never account for the majority of death. The vast majority of death in the world over the last 50 years has become chronic illness. Chronic disease accounts for all of it. And so this is really driving this home. So this is the NCD looking at worldwide deaths. More than 60% of all worldwide deaths are from chronic disorders. Only 30% are going to include things like the acute respiratory illnesses and things like that. And so it's always going to be 70% of mortality there. Even in the worst pandemics, they count for a very small amount of total mortality there. So in addition to kind of misunderstanding kind of the context of chronic disease versus acute disease, we've also misreported and misunderstood this concept of excess deaths in 2020. And so we've heard a lot about this excess mortality happening. At the beginning of this discussion, I laid out what I think is a really rational reason and good scientific underpinnings to a discovery that high PM2.5 from Australian and then subsequently California fires, combined with record high levels of hydrogen cyanide in the environment, tagged with a coronavirus that is able to traffic those things into the bloodstream, we had a setup for disaster. So we could have had a really, morbid event, massive amounts of death. But this is data from the CDC. I want to thank Scott Fulton for these incredible math that he's been doing for over the whole course of the year. He's been one of my sources of sanity, really, because he's been mining the CDC data over and over again to make sure that their data is adding up. And the data just hasn't been adding up to the common narrative over and over again. But this is looking at a very interesting phenomenon that we've been told that there's this high amount of excess death in 2020 compared to earlier trends. If we look at that trend over the last 7 years or so here, you see the annual death rate growing at 1.41% before this big catastrophic spike here in 2020. And in those years previous to it, you see something unique, which is kind of this steady on growth since 2015-16 of kind of a actually pretty remarkable growth, and then a sudden decrease. And so this first decrease in mortality, all cause, in the United States happened in 2019, just before the 2020 pandemic narrative. And so what I'm going to show you here is that if we had not had a low year of mortality here, there would have been a lot fewer elderly people right here to be prone to death this year. And so if I even this out for you and show you that if in fact we had not had a low year last year and we didn't have an increased elderly population that hadn't died last year, we would actually, if we had maintained a normal year last year, we would have seen potentially, a lesser death this year because they had died last year. So if you do the same number of deaths to keep up with that previous growth rate, which wasn't 1.41%, but was actually 1.9% when extrapolated over. So if we hadn't had this low year of 2019 and we had kept it going all the way through 2020 without this low year, we would have seen a 1.9% growth rate by the CDC numbers. In other words, we would see no excess death, in fact, at 2.94 million total deaths this year if we had seen the expected 2.88 million last year. So this is from CDC data. Looking at it for a shorter period of time, which I think is a more accurate way of doing this actually, is looking at the 20 weeks of the peak death that we should see from January through June each year. So from January to June, mortality here, looking at the 7-year trends, we again see a very low one in 2019, low death in 2019 as related to the total mortality, so total deaths here. So again, you see this posse of death in 2019 and then a catch-up year in 2020. So that not only for the whole year as a whole of 2020, but also very specific to this peak respiratory death rates that we saw in the first half of 2020. So we had a catch-up year, and the year before we'd had very low death in that first 6 months of 2019. And so when we say excess deaths and we're comparing it to 2019, we have to come to terms with the fact that we are accounting those deaths. If we keep it extrapolated across, whether it be the 6 months or as we previously showed you here for the whole year, both of them show a 1.9% year-on-year annual trend to increased death. Again, total death, not adjusted for population, or normalized for population, but not adjusted for age, is showing us this. So if we then go and were to start reporting this to the CDC as age adjusted as they do in Hong Kong, I think we would see a much flatter curve across the board. But nonetheless, I think that a lot of the excess death that's been reported is actually a function of the low mortality in 2019. This is a waterfall graph, another way of looking at the data, and it really kind of highlights above the midline there is excess deaths, and below the line is lower than expected deaths. And so you can imagine with that, the lowest mortality expected there since, in the last 6 years, there's an accumulated age. And in fact, in the NCHS data that comes up in a few minutes here, the average age of the population in the United States did go up on average by a tenth of a year. So there was an average aging over the course of 2019 because of that lesser than expected mortality in 2019, which left us with an older population that hadn't died the year before at risk for whatever would happen this year. And of course, in 2020, we saw the fires, we saw the cyanide, we saw all these things. And so the perfect story happened on top of what we would call kind of that high-risk population now that could have died last year, didn't, and is now more likely to die this year. Again, I want to point out that we see mortality really pretty stable throughout 2020 in a lot of different categories, but also ticking up in interesting areas across the board. So we said that there's been this many, some sort of 100 and 105 deaths per 100,000 individuals in the United States, and so pretty high mortality here. But even if this is right, and so far we've laid out that very few people could possibly be dying directly of the virus. But let's go ahead and pretend for a moment that all of these deaths are due directly to coronavirus. So coronavirus causes all these deaths. At the same time, you start to add up all the other comorbidities that led to mortality, and you see that coronavirus is going to end up being a very tiny portion of that whole. It's going to be a small percentage of that 30% of the mortality that's due to acute rather than chronic causes. Interestingly, as we would expect from a poisoning of the planet through PM 2.5, we see an uptick in the predictable vascular and metabolic conditions that would be poisoned by that, which would include the mitochondria in relation to diabetes, which increased 22.3% in 2020 unexpectedly compared to the baseline of the last 5 years. And again, a huge uptake in neurologic conditions, Alzheimer's, Parkinson's being among them, 16.3% increase. Stroke increased by 10%. Cardiovascular disease as a whole increased by 7%. And so in the conditions that would be most directly affected by PM 2.5 and toxin poisoning, we saw the uptick. So it wasn't just respiratory death and pneumonia or something like that. In fact, pneumonia as a whole decreased in 2020, which is pretty fascinating, right? And so whatever these patients are dying from right here in COVID-19 didn't show up as excess pneumonia death, which I find very fascinating. And so we need to take a close look at what really happened. It does not appear to have been a lung condition. It appears to be something that would have directly affected mitochondrial metabolism and vascular health that poisoned the planet in the last 12 to 24 months. I do want to come back to this just for a moment again and just emphasize how little we know, right? All right, so again, looking at these blue bars of mortality every single week or month here over the course of each year. So you've got these blue bars coming up, and we only account for this small amount of it as a specific virus, which means that the vast majority, 99%, goes unknown. And so for us to come in with any new PCR in the next couple of years and say that there's a whole bunch of new mortality, you need to keep in mind that it's probably the function of us starting to define what are these other viruses that are contributing to this high respiratory mortality in regards to the dysfunction of the innate immune system in this elderly population and this aging population. And so as we get increased population, both by age as well as total number, we expect mortality to go up over time. And we expect over time, as we define more and more viral traits and we get more and more genomics on viruses and everything else, we're going to get better and better at pinpointing some, or probably a tiny portion really, but a small portion of the viruses that are responsible every day of being in this healthy or unhealthy relationship with the innate immune system. And so we need to stop looking for the next virus and come to terms with, we don't know what causes the vast majority of respiratory death. And just because we named one and went out and did radical testing with PCR this year doesn't mean it was new, doesn't mean that the phenomenon was new. Excess death happened. The excess death was small compared to the trends, age adjusted, population adjusted. Another way of looking at this, again, just want to keep pointing out that the United States knows this stuff that I'm saying. I am not the only scientist saying this. I am not the only physician saying it. I'm not the one calculating these numbers. Other PhDs and MDs are finding this data and they are publishing it. This one is in fact from the NCHS, the mortality data. So the government agency tasked with keeping track of this stuff publishes just a couple months ago, December 2020, from Kenneth and Jishen Zhu and Elizabeth here, three masters in science, MD, PhD. They come together and they put this together of excess mortality or the decrease in mortality in 2019. So they're showing this phenomenon that we had a year of pause and we were going to have a year of catch-up mortality to be expected. So what they showed, life expectancy for US population in 2019 went from 78.8 in 2018 to 78.9 in the 2019. So there was an aging of the population, and the age-adjusted death rate increased by 1.2%. I'm sorry, decreased by 1.2%. So 1.2% less death rate per 100,000 individuals was a significant decline, and you saw it in the waterfall graph, et cetera, from the CDC. Now this is a different agency here at the NCHS, also recognizing this low mortality in 2019, setting us up for a high mortality event in 2020. What I did here for you is extrapolate all these numbers for you by the math. And so by in 2019, based on increase in population and age, we should have seen, we did see 2,367,000 deaths. This was 27,000 deaths likely to occur in 2020 if no change in death rate occurred. What I'm saying here is that this decrease from 395 million here to 395,000 to 367,000 deaths here. So there was a pent up of 27,000 decrease from the previous year. And so that we could have expected an extra 27,000 people that would have died but didn't die. But that would only be if every year was the same, when of course it's not, because what we've been showing both Hong Kong and the United States is those rates have been growing over time. So if you go ahead and show where we really should have been, expected deaths should have been up at 2.44 million, and we only had 2.367. So our actual pent-up demand for death was 73,000 extra deaths that didn't occur there. If you then continue that 1.9% growth 2018, 19, 20, we should have seen somewhere around 2.4 million deaths in 2020, plus the 73,000 deaths from 2019 that didn't happen, and we would have ended up around 2.56 million deaths. If you do put that all together, we end up with really no excess whatsoever. We could expect 192,000 additional deaths over 2019, which end up with excess there. The 10 leading causes of death in 2019 remained the same as in 2018, although the kidney disease and the 8th leading cause of influenza pneumonia and the 9th in 2019 switched ranks. So what that last bullet point is to say is nothing else really changed over that course of that 2019 year, 2018-19 year to suggest that there was any specific reason. So it was like one thing decreased. All of the death rates across all the disease types simply shifted down for a year. So by coincidence or less toxicity or whatever was going on, we had a good year. We caught up in 2020, and in the end, we could have seen an excess of 200,000 deaths without any change in our public health stats. So when you start to see 400,000 people died from coronavirus, but our total excess deaths are closer to the 200 to 300,000 range, even in worst case scenario, and then you have to take away 192,000, you realize we've had a slight bump, if any, in the total deaths this year. Our public health response, what have we done? So we've had a tough year. We've had coronavirus, we've had people who have behaved as poisonings, we've had people who behave as mild infections, we've had the whole gamut. It's been a tough year, but our response is terrifying to me. Far more dangerous, and I think far bigger mistake than we've made in any of our public health statistics or anything else, has been our public health response. Because of this narrative of this terrifying germ that was attacking us, this virus that was coming out of nowhere, that was coming out of China, blah, 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 this is how we responded. These are pictures of trucks spraying biocides up and down the streets of Europe and Italy and the like. This is pictures up in the right-hand corner of literally hazmat suited guys with dust blowers blowing biocides into the parks in Asia. We see biocides being sprayed on passengers as they come off of jet airplanes in Asia. We see agricultural spraying equipment repurposed for urban environments to spray biocides into this environment. Biocidal behavior of humanity is how this stuff developed. It comes down to obviously the hand sanitizers. Outside the United States, the same biocides used in the air are often used on the hands and in hospital settings, cleaning surfaces and the rest. Those biocides have now been outlawed just in the last couple of years in the United States, realizing that they cause so much cancer. And so the United States, a big effort from the EPA and the like came out to ban a lot of the hand sanitizers. And now we're down to pretty much alcohol-based hand sanitizers, because those that we were using before 2015 kind of time frame and those still used in most of the world, these biocides are potent carcinogens. And so I am really afraid of seeing what we're going to see in these coming years. We've already shown through my previous data and efforts that antibiotic and biocide behavior in the form of herbicides correlates very highly with our cancer death. And so high blue states spraying ends up with high death mortality in the red there. Flip the cancer map with the advent of the herbicides in the 1970s and 80s, but then with genetic modification of our crops in 2007 to 2011, we see the flipping of the map from the northeast, northwest to the south. And so we flipped our cancer map with the use of biocides in agriculture. Now we're using it in response to pandemics globally. I think we're going to see a huge uptick in the same cancers that we've seen go up with the herbicides. And these include leukemia, urinary bladder cancer on the right, and then down below here, one that's really going to jump, which is thyroid cancer. Already studies in nurses and the like that are exposed to biocides and their daily activities showed huge increase in thyroid cancer. And that was actually the data that got the EPA to start banning those biocides in US hospitals and other systems and kind of consumer level hand sanitizers and the like. And so we're going to see a huge uptake of thyroid cancer with this global intervention of biocide use. Liver and intrahepatic duct cancers are expected to go up as well. This is looking at senile dementias and its advent and rise with the biocides in agriculture. Again, I expect this to rise again. We've already got an epidemic of Alzheimer's and Parkinson's in our women and men respectively. We're going to see this worsen with this global biocide response. This, of course, is looking at a seemingly less benign thing, which is sleep disorder in relationship to herbicides and biocides. Unfortunately, as I showed you earlier, sleep disorder is one of the primary things to disrupt the innate immune system and put us in an adversarial or diminishing relationship with the microbiome at large. We then put masks on everybody. These masks are created with melt-blown plastic. This is polypropylene that is sprayed against a spinning screen, and a net of melted plastic, or these nanofibers, results. And then we put that inside of an artificial membrane mask, and we put that over our face, and we breathe through that. This is an electron microscope of these polypropylene fibers, and what you can see instantly is that they are very fragile and friable. They are falling apart, even coming right off of the mesh plastic screen that produces these masks. And so you can imagine all these little fibrils coming loose with simple flexing of the mask, or with heating and humidification through your breath, and you're now breathing polypropylene into the tiny little airways. These are tiny nanoparticles here at this level. So this is 100 microns at this size. So you're down at the nano size when you get these tiny little fibrils that are falling off the mask and being breathed into the lungs. This is a study done in 2005, that far back, showing the respiratory effects of occupational exposure to polypropylene flock, like I just showed you. And these work-related respiratory symptoms were reported in 26% of exposed individuals versus 13% in controlled manufacturing environment that didn't have the polypropylene exposure. And so as they did the logistics regression of that data, it turned out that there was a 3.6-fold 3x, almost 4x increase in respiratory symptoms in polypropylene producers over non-polypropylene producers. Forced vital capacity, forced expiratory volume of the lung, and forced mid-expiratory flow of the lung, and DLCO, which is the diffusion capacity of oxygen across the lung. All of those factors of lung function were decreased in the polypropylene manufacturing group. And so, or not necessarily manufacturing, but somehow occupationally exposed to the polypropylene flock. They demonstrated that increased IL-8 and TNF-alpha levels were increased, which is exactly what you would see as a predisposition to the inflammatory consequences of something like SARS-CoV-2 or the like. And so not only was the lung function diminished, there was vascular inflammation and immune dysfunction happening, predisposing multi-organ failure and hypoxic injuries just by being exposed to polypropylene. HRCT, which is resolution CT scans of these individuals, demonstrated that in a significant sum of the subjects, there was actually the ground glass attenuation found on CT scan, which is the exact same thing that we saw develop after our patients with SARS-CoV-2 were hospitalized. And so is it possible that we have SARS-CoV-2 demonstrating PM2.5 and cyanide toxicity to the body? They present with hypoxia, histotoxic hypoxia, and our immediate response is throw a polypropylene mask on them that they can't breathe well through, and they start inhaling polypropylene, which contributes to the diminished lung function and the increase of ground glass inflammation in the lung that was so indicative of the SARS events. We have to take a look at our behavior because we've proven that increased testing causes increased mortality, meaning we are doing things very early on in the management of this condition that is causing people to have worse outcomes. And I believe masks are one of those. Effective of N95 respiratory versus surgical masks, they've demonstrated in this review article that there was no increase to protection from viral transmission in hospital settings with the N95 mask, which we've been told is the gold standard. The N95 is a polypropylene mask versus the surgical masks, even the ones that are not melt-blown nano filters and all of that, supposedly less effective, have actually the same protective quality and probably less toxicity than these N95 melt-blown plastic masks that we are now distributing to so many people throughout the world. So fortunately, we are geniuses because we did not respond with a bunch of N95 masks. In the United States, I just have to take my hat off to the ingenuity of our population. The coffee filter is my favorite one that I've seen so far. The good old coffee filter repurposed as the coffee filter mask. I love the dog cone approach. I think this is underutilized in a lot of environments. I think we should all stop touching your face. Obviously, it's the whole purpose on a dog. So if the CDC is right, is that we propagate viruses by touching our face, this is probably more effective than and less toxic than N95 masks. And so with some humor here, recognize that tying plastic bags over your face is probably never recommended. But my favorite fact is that in my humor at the beginning of this thing, I saw this gentleman I took a picture of at a grocery store at the time, just amazed by the idiocy of like, oh my gosh, this guy, poor guy, really thinks he's doing something good by wearing a handkerchief over his face. In my medical training, that's ludicrous. That doesn't do anything whatsoever to stop the transmission of viruses. But in the end, after I've studied everything I've studied this year, I think the safest thing on this thing is the cool looking dude with his shades and the bandit mask on, because there's no polypropylene in that cotton handkerchief. And he's probably the safest of all of us, walking around looking cooler than the most of us as well. So we have to be impressed by the fact that we know so little and so much of our public health response is so potentially toxic. Nowhere is that more true than with isolation. And so isolation here is a terrifying single risk factor for poor mortality outcomes. This is taking a look at the risk behaviors that are typical of COVID-19 death. Risk factors like high blood pressure, high cholesterol, obesity, and the like, cigarette use, etc. What this is showing is in the most sedentary group, the most isolated rather, the most socially networked isolated group at 0 to 1 interaction levels have as much as 3-fold increases in risk factors of these behaviors over those that have high social index scores. And so social isolation as a single agent to increase the behaviors that increase the risk of COVID-19 death is what I'm showing you there. I love this social response. Everybody remembers Fauci coming out to say that nobody should ever shake hands again in the United States or the world. And so we have a new normal of never shaking hands. So they were coming up with fist bumping or elbow tapping and all of this at the White House, et cetera. And so in this humor, I just was dumbfounded because already I had found studies showing the opposite, that in fact, close contact, including hugging, are some of the most frequent ways to reduce your risk of respiratory infection. And so in these studies, many of them out there now, but this one, a good one in 2015, looking at multivariate analysis of risks and other things, showed that there was a 61% reduction in respiratory infections in those that got hugged frequently. And the highest rates were linear with the number of hugs. So those that got 7 hugs a day had even far less than the 61% of the general group that was getting at least hugged once a day. And so social contact, intimate nurture of one another has a profound effect on our innate immune system. That seems to be far in excess of any germ theory kind of interaction with the virus. And so in a bizarre way, we probably should embrace Fauci's statement of never another handshake and just make sure we hug each other from here on out. And so more hugs to decrease the toxicity of the environment. The real risk factors for COVID-19 mortality, this is a summary slide here. This is really what we should come away from, that microbiome stress in the soil, water, and air systems and loss of biodiversity for our soil, food, water, and air is where we begin these unhealthy relationships with the virome, where the virome has to make adaptive extinction level stress decisions and adaptations that we are then needing from that virome to update our own biology for a more resilient body. So destruction of the microbiome was the first modifiable risk factor for the future prevention of pandemics. Second one, glyphosate and similar herbicide biocides in our environment have to be stopped. If we do, we will have a dramatic reduction in the seasonal and total burden of respiratory disease as a mechanism of mortality in humans. The aging population is definitely going to continue to increase our risk and vulnerability to these things. We should not be shocked. We should not be told there's excess deaths until it's been age and population adjusted. So please ask your politicians and your agencies for the right data. We need age and population adjusted mortality rates in regards to pandemics or epidemics or endemic disease or whatever it is. Age and population adjusted. We need it. High comorbidity rates in chronic disease is the pandemic. We are not seeing a pandemic of infection. We're seeing a pandemic of vulnerability. And the primary risk factors, the primary vulnerabilities are the chronic diseases. High prevalence of drug treatment with ACE inhibitors and statin drugs for all of those top 5 comorbidities. These are the things we need to be alert to prevent the next coronavirus pandemic. Vitamin D deficiency, we need to be on this. We need to be treating that aggressively. Influenza vaccination, if there's word of a coronavirus, we need to stop flu vaccines for that year or two until that coronavirus goes through the populations more safely. Fear, we need to stop the fear. We need to address who's at high risk. We could use PCR to, again, differentiate who's at high risk and how are we going to manage them differently to mitigate those risks and reduce mortality and suffering. And more than anything, stop the isolation. It's time for a 3-dimensional understanding of the world around us is really the message I'm hoping to drive home to all of you today. I know this has gone long today. I know I've thrown an enormous amount of science at you. I'm kind of shocked to see that there's anybody left on here. As I bump back to the screen, still see 4,000 of you on here. You guys are troopers. You got a gold star already for hanging in there this long. But I want to kind of bring home some final thoughts for you here. We need a 3-dimensional understanding of human biology, and we need this transformation to happen fast, right? We need to quickly come to terms with the fact that we are a small part of biology. We are a part of an expression of biology on the planet at large. And in that expression, we have the opportunity, because of our conscious capacity, to witness the sheer beauty around us and participate in it rather than war against it. I feel a great urgency to bring some peace to the environment that we find ourselves in today. We need to look towards healthy pregnancy, birth, childhood, adulthood, and the wisdom years. We need to revere our elderly. We need to give them more hugs. We need to be engaged at every age, from our children to our elders, and we need to learn from them, and their engagement in the greater world around them is going to be critical to our future generations and their propensity towards pandemic or resiliency. And so these are the steps we need to take. We need to stop the war on the microbiome. We need to stop the biocides. We need to stop the herbicides. We need to stop trying to kill everything. When we wake up in the morning and our first thoughts are, what are we going to kill? We're going to die. We are literally marching towards that extinction event. So we need to instead wake up and instead consider perhaps what is the beauty we're going to participate in. So I can foresee the possibility of changing our social structure, our homes, our communities, to get outside, breathe your biome, as we've said so many times, get engaged with a diverse ecosystem, touch as many viruses and bacteria as you possibly can, eat and drink from water systems and soil systems that are still intact, which is becoming so difficult to find on the planet today. We need to go beyond organic agriculture. We need to go to true regenerative agriculture that makes the point of agriculture not the crops that we produce, but the health of the soil, the vitality of the soil, the volume of the soil. We need soil farmers, not crop farmers. We need to nurture this earth back into its full capacity to deal with carbon in the atmosphere. We need to pull all that carbon out, not seasonally, but permanently. We need to get the carbon balance back on this planet, or we will continue to die of worse and worse pandemics as the carbon continues to accumulate in the atmosphere at the dead end as we destroy 97% of the arable soils around the world. We need to engage nature. We need to stop running from her. We need to get engaged with proactive efforts towards prevention. I do want to just show you one methodology that you could adopt today. In addition to getting outdoors, we're in the middle of winter in the Northern Hemisphere, and vitamin D is an incredibly effective treatment mechanism, as I told you. I showed you the risk of morbidity and mortality, but I also want to show you for a moment the incredible impact of, let me see if I can share this here for you, because this study is worth seeing for a second. Maybe it's just not going to let me. This may be as tired as you are right now, but here we go from current slide. And so this is looking at the vitamin D treatment mechanism. This was just published in October of 2020, demonstrating the treatment with vitamin D of acute hospitalized COVID-19 patients. So I really want you guys to take this to heart, because if there's one supplement you're going to add for an innate immune system outside of the ion biome stuff, is going to be vitamin D. And so ion is the product that I've showed you too many times. But if you need to go back to the ionbiome.com website, go do that and figure out how it helps support the rebuilding of the barrier systems, the tight junction barriers of the blood-brain barrier, the gut barrier, the kidney tubules, all of that. So we got to get the barriers back up, but then we need to accelerate the innate immune system's function behind that barrier. And that's what vitamin D does. So in this treatment demonstration, this was a randomized trial with hydroxychloroquine, which again is treating the toxicity side of the equation, while the vitamin D is changing the relationship to the virome. And so hydroxychloroquine improving oxygenation, and then vitamin D improving the innate immune system. In those treated, 50 patients treated with vitamin D in conjunction with the hydrochloroquine, only one patient died. And that was for a total of 2% there. Of the people that did not get treated, they picked 26 patients randomly not to be treated with vitamin D. They actually had a mortality rate of 50%, 13 of the 26. I'm sorry, they had an ICU occurrence of 50%, and 2 of them died there. And so they reduced ICU, there you go. They reduced the ICU from 13 down to 1 with vitamin D treatment. And so if there's one other thing to do beyond the barriers, it's the vitamin D therapy. And so I recommend somewhere around 2,000 to 5,000 IU daily, depending on the age. Children obviously lower, adults somewhere in that 5,000 to 10,000 units a day, typically in our clinic. But for you, I would recommend you touch base with your medical practitioners. I think it's a very good idea to get vitamin D tested across your family and get everybody up to a level of 50 to 80 units per deciliter. And so those international units at 50 to 80 rather than the current normal that will be reported on the lab is 20 to 80 is normal. I really want it up at that 50 to 80 level for my patients and for my family. So get your vitamin D level checked. Consider getting on that as a frontline defense mechanism to changing your innate immunity. The ion biome to support the gut linings is obviously a very biased opinion there with our lab cranking on that data for the last 10 years. But those are some things that I think you could really take advantage of. In the end, I think that this has been a speed bump. COVID-19 has been a minor speed bump in this incredible extinction event journey that we are heading towards. If we continue with the current mechanisms of response in the public with isolation and gloving and masking and biocides and all the rest, we are on route to see much greater mortality and morbidity around us. Can you imagine what it's going to look like when we actually start to shrink the population with these horrific potentials of disparate relationship to the virome and the microbiome? Mother Nature is just. She has the tipping apparatus to make sure that if we misbehave and we radically abuse her, we will die. And she has made our health integrated into her very fabric of the microbiome and the virome. If we can't come to terms with that, we will put ourselves into a death spiral. So I want to conclude. I'm thankful for all of you being on. I want to conclude with a few thoughts for populations. For all of you getting outside, I appreciate you. But for those of you who are scientists and physicians in this group, some of this should raise questions. I don't pretend like I have all the answers. I don't pretend like my data or Scott's data or any of the other scientists that are involved in this data have finished the puzzle. We want science to progress. I would call you into action as a scientist, as a physician, to think critically about our relationship to the ecosystem and start to help me ask better questions about how do we get our children in line with nature? How do we get aligned back up so that we rewrite human biology into Mother Nature herself? For those of you that are part of the current effort towards the vaccines and everything else, I would encourage you to consider what's about to happen. We are about to genetically modify the human species in response to, it looks like, a very minimal excess death event. And once we start to really look at the overall picture, age adjusted, population adjusted, and adjusted for the low 2019 mortality, we realized 2020 was really not a remarkable year at all. From a public health standpoint, I would say that we are looking at a relatively benign year, and we're making catastrophic and irreversible decisions to make medical normalcy out of genetic modifications of humans through this mechanisms of RNA and DNA transplant via viral and other transfection mechanisms that we're calling vaccines. We have never had vaccines that genetically modify human beings. We do today. And these three organizations, AstraZeneca, Pfizer, and the like, that are rolling these out are a tip of the iceberg. I think there's around 72 different vaccines, genetic vaccines, that are in manufacturing and testing at some point now. And so we are going to see hundreds and hundreds of these come out over the next couple of years. And we have just opened up permission for the government and health agencies to demand genetic modification for one virus. There are 10 to the 31 other viruses out there. What is the next one that they're going to demand our genetic modification to? They cannot stem the tide of 10 to the 31 viruses in the air that we breathe. They're going to stay there, and they're going to stay in relationship to biology on Earth, human or otherwise. And if our relationship is adversarial, we will die. And so please help me imagine a different future of vaccines. I have a request for you to go on our change.org website. If you go to the Global Health Education section of the Zach Bush MD website, you'll see the call to action for the child health immunity and vaccination future, where we're going to start asking it. So if you sign that petition for us to start to be able to take to the government to get funds allocated towards looking at child health immunity and vaccination in the context of the new science of the microbiome and the virome, we need to update this. It is no longer the Salk Institute science of 100 years ago. We know that these viruses and the microbiome at large are part of our future if we're going to be resilient, if we're going to avoid extinction. So please join me to sign that petition. We need a different philosophy around vaccination. We need to rethink vaccines in the context of the microbiome and the virome. We don't need to eliminate vaccines. We need safe vaccines. We need to stop doing things that we know are not safe. And we need to start a different approach to dealing with childhood immunity through asking different questions. So join me there, change.org, or through the Zach Bush MD website and the Global Health Education group. To all of you, I want to thank you for your participation in this Global Health Initiative. You guys have supported this. The only funds are being donated by you that help us get these pieces of information out there to you. If you haven't consumed the virome, sleep, and the innate immunity sections that we've done previously, please dive in there. It will help you understand today's content much more vividly. And so thank you for your participation, your patience. Dr. Peter Cummings, thank you for your participation in answering the Q&A as it's come through. We have 891 questions. I trust we've been unable to answer every single one of those, but Peter's been trying to keep up his best through the course of this, and we will continue to put out an effort in the coming weeks to expand the resources that will accompany the recording of this. In addition to the recording with all the slides that you've seen, I'll also have a huge reference library of all of the science, some of which has been included in this, as well as many other documents that I've pulled over the last year that help us with this bigger understanding of the events of the last year and what's happened there. So continue to revisit over the next week or two the Global Health Education event on the Zach Bush MD website for more of those resources. I would ask that you share this with as many people as you can, in the sense that we make this free for the purpose of getting this information out there as far as we can to make the public health impact that we need for the future of our species. And so if you could do me a favor in return for the efforts that I've put forward today and in the many months previous in preparation for this, please get the word out there. Please help enrich the public narrative. It really hurts me at the soul level to hear the common narrative being pitched around moment by moment, day by day on the airplanes I fly on and everything else. I just hear nauseating and narrow-sighted and short-sighted belief becoming gospel for so many people, and that religious-like fervor that is currently around the current narrative is terrifying. If in fact we see criminalization of anti-vaxxers and the like out there, I will mourn that day for modern science. There should be no such thing as science dogma. The moment we stop questioning each other, the moment you stop questioning me, is the moment science dies. The march towards the understanding of knowledge that science is requires a billion minds, and we're so lucky to have 7.8 billion brilliant minds on this planet today. And if empowered to go ask the questions that make sense to their environment, instead of a dogmatic and paternalistic, one size fits all narrative, yes, no, you have COVID, therefore you might die. That kind of dogma needs to end. We need to respect the consumer. Each of you are so intelligent that you would even tune into something like this to keep up with 3 hours of content like that. You're near crazy. So we need to respect that kind of crazy curiosity and intuition in the public, and we need to go after that. So last thought, I guess, for you, because again, I don't want to end on a fearful note. If you are feeling unstable and fearful from the knowledge that we've shared today, and you feeling that you do not have a healthcare team around you that has a holistic approach, you feel free to reach out to the Intrinsic Health series, which is available, links at the Zach Bush MD website, as well as through the other pathways that I've mentioned earlier. But the zachbushmd.com website will get you to the Intrinsic Health series. It's an 8-week program, one-on-one with a coach, or a group coaching for 4 weeks program there. And so you got 2 options, 2 price points for a real deep dive on everything we do in clinic to change the innate immune system, change our relationship to the virome, and get a resilient biology built back in you. So the Intrinsic Health series, you can access it anywhere in the world. We have participants from every Western country really at this point, as well as some Eastern countries and South America. And so it's just really a joy to see an international community coming in around the Intrinsic Health series there. If you need more desperate and more comprehensive health because of health crisis you may be in, then our clinic is available to you. themclinic.com can get you to that team. We're eager to support you with our multi-specialty group there, Chinese medicine all the way to modern medicine, to understand how to put together a resource pool around you that understands your relationship to your biology at large, the ecosystems around you, and the microbiology within you. So we'll try to continue to roll out more and more resources. Stay tuned with zachbushmd.com. You can sign up for the monthly blog. Dr. Peter Cummings prepared an incredible one for this event today. If you haven't taken a deep dive on his, it is a must read, because he goes into the details on death certificates and the common errors that we make as physicians that then give a very erroneous picture to the public health officials that are collecting that data. And we can be really part and parcel to the mistakes made as physicians filling out that paperwork. And so I think it's an interesting deep dive there with Dr. Peter Cummings on the blog. So please tune back into that. You can tune into the newsletter for frequent updates, and of course, social media, Facebook and Instagram, Zach Bush MD, you'll find us there. We try to keep pumping out the good stuff because I'm always as determined to make you hopeful for the future as I am to raise the warning flag of the wrong directions we may be going in. So thank you all. So much gratitude to each of you. Really can't say enough to the encouragement that is to me to see so many, over 20,000 of you joining this project today. So thank you for that. And I look forward to the ripple effects through the millions of consumers that you touch and the family members and beyond. So thank you. I honor you. I appreciate your diligence and your attention, and best of health to you and yours.